Okay. So, hi guys. Today we're going to go over all of the post-midterm material for immunology. If you guys have any questions, feel free to stop me at any time, all right? Or just type in the chat. I'd prefer if you guys just like unmute yourselves. It's easier to catch up, okay? Tamam, let's start. All right. So, those are your beloved cells, the T cells and the B cells. So you know, guys, how it's like choose your character. Da, da, da. So that's the idea behind the slide. Okay. Can you guys give me some characteristics of T cells and B cells that come out of your mind, like the top of your mind? Just anything. When you hear T cell, what's the first thing you think about? Or B cell? Okay.
T cell would kill the antigen by itself.
Okay. Okay. Interesting. Tamam. And B cells would secrete what? It would secrete antibodies, right? هلا T cells and B cells كمان they reside in different places in the lymph node. So let's talk about the lymph node together. So for the lymph node, you know that the lymph node is basically we have a lot of lymph nodes all over our body. It's a place, it's like an airport. It's basically a place where all of the blood, the lymph, everything passes through it. So it kind of keeps our body under surveillance from any antigens, any bacteria, anything that could harm our body, basically. All right. And if you guys look at this lymph node together, we can see that antigens usually like they make up their way there because as I said, it's like an airport, everything goes through it, but not everything leaves. It kind of holds into the bad stuff. Okay. So if we look together, let's start with here. So the B cells and T cells together will enter the paracortical region or the parafollicular region. And in this parafollicular region, basically the T cells reside. All right. So whenever you hear parafollicular, just know it's T cells. And then the B cells are not going to stay there. They're going to leave behind the T cells and they'll go to the cortical region, all right, at the peripheries. So the B cells are going to go to the cortical region and they're going to form something called the primary follicle, all right? This primary follicle is just basically a cluster of B cells that are residing in the cortical region. Now, if the B cells that are there, no, you did not miss a lot. I just started. Anyways, if the B cells that are in the cortical region that formed the primary follicle, if they get activated, all right, by an antigen, by T cells, whatever, they will form the germinal center. And if you guys, you can think of it this way, germinal center, germinal means
هو نايم
okay, so the germinal center is just something that's growing. So since they're activated, they're kind of like multiplying, and they form this germinal center. And usually this is a secondary lymphoid follicle. Why? Because the primary follicle that we talked about right here is just created by the presence of B cells. But if they get activated and they form a germinal center, this is a secondary follicle. All right? These are just basic anatomy. I'm just like easing you guys into it. All right. So we said and note the lymph node as like an airport, and everything goes into there, and you kind of can think of it as well as like a filter. It filters everything in the blood. All right. So usually antigens are going to go in there. All right. And the most important thing you have to know about cell interactions between B and T cells is that when an antigen goes, all right, so the antigen is going to go into the lymph node. This is a lymph node, all right. What happens is the antigen is going to meet both. It will meet the T cell and the B cell, all right? And it's going to activate both the B cell and the T cell. So if you guys look in here, we have one here and one here, all right? This is the one right here is the T cell, and in here we're the B cell. And you guys can see this is the basically the parafollicular cortex, and here it's the follicular, like the follicle. All right. Now, dendritic cells or any antigen presenting cell is going, so we said, the antigen will go into the lymph node, right? So it's not going to directly go into the cell. Basically, dendritic cells and macrophages that are inside the lymph node, they're going to pick up this what is it called, antigen, because you can think of them as like they just clean around like a vacuum. So they're going to like suck up anything that could be like an antigen. And what will they do? They're going to present it on their surface. All right. So a naive T cell, naive means it's still a baby, it did not grow up, it's still undifferentiated, is going to interact with the dendritic cell. And this is going to cause T cell activation. And in here specifically, when we're talking about B and T cell interactions, you guys have to keep in mind that this T cell that is the most important in this interaction is basically the helper T cell, right? So what happens is after the dendritic cell and the T cell, they interact together, we're going to have T cell activation forming helper T cells, okay? And I said that simultaneously, the B cells are going to get activated. And if you guys look in here, okay, so this is a haptin carrier, all right? So basically it's like a protein and then it has the antigen to it. And what happens is the B cell is gonna kind of take the, like it's like a protein that carries the antigen, all right? It's going to like kind of read the antigen, it's going to take it in, all right? And then it will cause the formation of, or like it will cause the B cell to express an extra receptor. And what is this receptor, guys? It's the CD40. And you can guys see it here. Like if you look at the normal B cells, they just like not the normal, I mean like the naive B cells, the not activated, they're just there hanging out without anything out, like without anything poking out of it, all right? So after the B cell is activated and the T cell is activated, they're gonna need to interact. And you can think of it and you're like, why do they need to interact? We just said that they're activated. They can go just like, like just go and like kind of fight the infection on their own. Why do they need to interact? Well, the activation of the B cell is the most effective after being basically kind of even more charged by the helper T cell, okay? So we said that the B cell basically expressed the CD40. What will it do? It will go and it will try to meet the T cell. طبعا, how will it know where to meet the T cell? Basically, when all of this is happening, chemokines are being released. Okay, and we know that chemokines, what are they? They're chemical attractants, so they're calling on people. And those chemokines, they are extremely specific to those activated cells. So these, like you have to guys imagine with me that we have chemokines like right here. Okay, wait, no, I don't like this. You have to like imagine with me that we have like chemokines all over the place in here, all right? These chemokines are kind of calling upon the T helper cell and the B cell. So they're going to follow the chemical signal and they're going to meet in here, all right? Once they meet, that's when the CD40 on the B cell is going to interact with the CD40L on the T cell. And once this happens, basically, this is going to give the B cell the boost we need to go and fight off the infection. How? By basically, it's going to trigger the formation of plasmablasts and memory cells. Okay? What are plasmablasts? Plasmablasts are basically the type of B cells that secrete antibodies. All right? So after this interaction happens and they already have the like antigen, they already kind of understood like how the antigen looks and what's the structure. So they like the B cell only needs a little bit of more help, a push from the T cells to start forming antibodies that are against this antigen. Okay, and that's what happens. So after this CD40L and CD40 interaction, we're going to have, it will kickstart the formation of plasmablasts that will start forming the antibodies. all right? That will basically attack the the thing that is going on. Now we're going to touch upon this a lot, so don't worry if you don't get it now. We're going to talk more about it as we go on, all right? Is everything clear up till now? So I'll do a very fast recap, all right? So what happens? We have an antigen. This antigen is going to activate both T cell and B cell, all right? Once the T cell is activated, it's going to change into something called a T helper cell, specifically a T follicular helper cell. And why T follicular? Because we are in the follicles, okay? Or not we are in them, but we want to help the B cells that are usually located in the follicles, right? And then the B cell is going to be activated, but it's going to need more of a push from the helper cell, or it needs a little bit more help to kind of boost and kickstart the whole plasmablast formation, the memory B cells formation. All right. So what happens is we're going to have chemokines that will attract the activated T cell and the activated B cell, where the interaction of CD40 with CD40L ligand will happen. And this interaction is so important. Can someone tell me why? There are two processes that are very important and related to this interaction between CD40 and CD40L. Yes?
We're gonna activate the T helper cells, the naive T cells, into making them cytotoxic T cells, and the B cell, we're gonna make it more activated. Basically, we're gonna make the B cell plasma cell, I guess.
Okay, this is you're right. The basically for the B cells, they're going to be activated to form the plasma cells. And more than that, we're trying to, the most important thing is in here, when we want to create a plasma cell, the plasma cells, we said it's going to secrete antibodies, right? We
Yes.
want to ensure that these antibodies that are secreted by the B cells are high-end quality, they're top-notch. Why?
Yes.
So they can go and attack the thing that caused all of this from the start. All right?
Yeah.
Now let's talk more about it as we go on. Tamam?
Yes.
Oh, wait. Sorry. Okay.
So the interaction would be between B cell by CD14, right?
CD40L, like the ligand, all right? So
And
basically,
the T, which ligand would be on the T?
wait, okay. On the T, we're gonna have the CD40 ligand. On the B cell, we have the CD40 receptor that's going to interact with the ligand. All right?
Oh, okay. Yeah,
You can
makes sense.
see
Thank
it
you.
like this is the receptor right here. Come on.
Yes.
Okay.
Before I start, when the antigen binds to the naive CD4, it's going to become helper T cell.
Yes.
And the helper T cell will become follicular T cell, which is going to bind to the plasma, to the B cell, so that it can further activate it.
Yes, exactly.
Okay, so basically in T cell, we're only going to need the antigen to activate it, basically.
Yeah, in this case, yeah, we're going to talk about other activations later on. But
Yeah.
in this specific case, we only need the antigen to activate the CD, the naive CD4 T cell to form helper cells. All right?
And the plasma antigen, and then it's going to bind to the initial T cells.
Yes.
Okay, thank you.
You're welcome. So this is just everything that we said, but on text, all right? So we said that the T helper cells provide the crucial help through this interaction, okay? So without this interaction, we're not going to be able to kickstart the B cells to fully differentiate into the plasma cells and the memory B cells that we need, all right? This is everything that we talked about. Now, why is we said there's other reasons for the CD40L other than the interaction. We said that it's not only to form the plasma cells, but come on, we have to think about what does these plasma cells do. These plasma cells, they're going to secrete antibodies. These antibodies that are secreted, we want to make sure that they are, as I said, top notch. They're amazing, why? Because if you just create whatever antibodies, do you think it's going to be effective? No, we want them to be highly specific and highly attracted with high affinity to the like the antigen, all right, or like whatever caused this antigen to be released. تمام. So CD40L and CD40 is going to trigger isotype switching and affinity maturation of B cells. Okay. And when we say isotype switching, does anyone know what isotype switching is?
We're trying to make variation in the antibodies so that it becomes more specific.
Okay, true. And this is more of affinity maturation, but the isotype switching is similar to what you said, but we're trying to change them from basically exactly IgM to IgG. Exactly. So and what Muhammad said is right, because IgG is more specific. IgM is more like, it's more
Uh, First line defense.
yeah, exactly. So basically IgM is there everywhere, but IgG usually is more specific. So let's see. Just 1 second. I don't know how to stop. Okay, like that. Perfect. Um, Remember, guys, this is just to remind you of the hapten carrier. You saw in the beginning when I said that basically the hapten is attached to a protein, and this helps basically the B cell to be able to present it on its surface. You get me? Like this is just a like reminder of everything. So basically, we said no. Basically, we have the CD40 and CD40L between the two cells. But كمان we have another thing, which is, you see how like the antigen is presented on MHC class 2. So come on, there will be like another interaction where the MHC class 2 is gonna come on, like basically attract the T cell. So the T cell is going to be attracted by two, like we're going to have kind of two interactions, okay? MHC class 2 with the antigen, like that has the antigen in the B cell with a receptor from the T cell, and the CD40 to CD40 ligand. But the most important one that we need to know is the CD40 to the CD40L, which is the ligand, all right? Now, later on, we're going to talk about vaccines. The hapten carrier, or basically having like an antigen attached to a protein, basically causes is another name for conjugate vaccines. Okay, like when they create vaccines, sometimes they do that. They put an antigen to a carrier protein just so they like stimulate a stronger immune response. Okay, and this is all you need to know from here. Tamam? Let's talk about the isotype switching. Okay. For isotype switching, we said that it's triggered by the CD40 and the CD40L. You see? So basically the ligand is on the T cell, the CD40 is on the B cell. Once they interact, isotype switching will happen. So why will the isotype switching happen? Because this interaction is going to activate this cute enzyme called AID, okay? The activation induced deaminase. What will this enzyme do? So you see, guys, I'm pretty sure you know the structure of basically the IG, like the receptors, all right? Like the IgE, IgG, so on. So we're going to have a constant region right here, and we're going to have a variable region. Okay, now this constant region, you can see it starts with a C, so constant. Now, what happens in isotype switching is that the AID that is activated by the interaction between the CD40L and the CD40 is going to basically cause variation in the constant. So you see the constant. So what's changing is the constant, like the constant chain is what's changing, and the variable is gonna like just be the way it is. And this is going to kind of, let's see. So this is going to, like, this enzyme is going to work on the constant. It will break some things up. It will, I don't know, it will rearrange, it will do its magic. And then we're going to end up with something like that. Like, see how it changed from this long chain into this tiny chain. So this is going to make the IgM change into IgG, all right, which is more specific to the like microbe or whatever caused the antigen to be released. Okay, is this clear? You can guys just type in saying clear. Okay, perfect. Now, another thing that we said is basically kind of affected by or is influenced by this interaction between CD40 and CD40L is something called affinity maturation and somatic hypermutation. It sounds complicated, but it's so easy, guys. It's just the concept that the antibodies or the antibodies that we created, or the antibodies we created, these receptors that you see, we want them to be, as I said earlier, top notch. Now, how do I make sure they're top notch? We want the affinity to be so strong. So in case the microbe was kind of hiding, it will still sense it and it will go and attach to it, and the whole attack will happen. Okay, so how does this happen? So it happens, so when you say affinity maturation, we're kind of helping the affinity to mature, become stronger and stronger. All right, it's like it's going to the gym and it's like building up some muscles. So how does that happen? It happens through something called somatic hypermutation. Somatic hypermutation are basically mutations that happen in the variable regions. So see the difference? In the isotype switching, the change happens in the constant region, while in here in somatic hypermutation, the mutations happen usually or cluster in the variable regions. And these mutations, you can guys think of it this way: mutations, let's think of mutations as like bad patches in life, or something that's challenging. So what happens is, as this cell goes through more challenges, so it's going through more mutations and more mutations, more challenges, what happens is it's going to gain more and more strength. So these somatic hypermutations are going to increase the affinity maturation, resulting in strong antibodies. All right? So in here, you can see that more mutations occur in IgG than in IgM, because if you think of it, IgG is more specific, has higher affinity to what we need than IgM antibodies. So because of that, obviously it's going to go through more hardships in life or mutations. Okay? طبعا all of this has to do with like AID. Okay? Now, we know that we have multiple different kinds of antibodies. We have IgE, IgG, IgA, right? So we have something called cytokines, or basically chemokines, all right? Or like specific chemical mediators that can provoke or stimulate the formation of specific antibodies, okay? So the most important ones that Dr. Garwin mentioned were interleukin 4 and interleukin 5, and there's interleukin 13 too. Okay, and I'll tell you guys how I memorized it. So basically it's like 4, 5, 13. Basically in my head it's like 3, 4, 5, but the 3 comes at the end, and it's like 2. Okay, now I complicated it. Just never mind. But that's how I memorized it in my head. In my head, it's like you can think of it 2, 3, 4, 5. 2 stands for like 2 is close to the 3, all right? And because the 2 is close to the 3, this means that 3 is made up of 2 digits, which is 13, because I always used to mix it up. And then 4 and 5. So 4, 5, 13. Okay? You can ignore that. It's just the way I think. Anyways, so this is a very important detail. Why? Because imagine even later on in your step 1, they love asking these questions. Like they love interleukin 4 and 5, and like the idea that interleukin 4 and 5, if they're there, they might result in the formation of IgE. Okay, now another like chemical mediator, interferon gamma. When we have interferon gamma, you can think of it, gamma starts with a G, so IgG, all right? So interferon gamma is going to stimulate the release of IgG, all right? And then we have TGF beta, okay? For TGF beta, it's going to stimulate the cell to release like IgA, okay? And for TGF beta, guys, I have a question. And if you know how to answer this, I'll tell you what I just thought of. What is it called? IgA, where is it located the most?
Mucosal membrane. Basically, yeah,
Exactly.
the
So
mucosal membrane.
IgA usually lines everything that's mucosal. And when you say mucosal, what do you think of? You think of your GI, at least for
Mhm.
me. Okay. So do you know Fridays, the restaurant?
Yes.
Okay. So do you know how they usually like they write it TGIF?
I don't know. I don't really go to.
Okay. So basically, this
Yeah,
restaurant.
that stands for thank God it's Friday.
Yeah. So basically, TG, you can think of it that way. So something TGF is like TGIF and it's food. So food means mucosal. So IgA. Okay, like I'm just trying to create like stupid like 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 things to link together. تمام؟ فعنا interleukin 4, 5 and 13 for IgE. عنا interferon gamma G so IgG and TGF beta IgA. Or another thing, it's like it ends with a beta with a B يعني and AB, you know? I don't know, guys. I don't know if you guys think the same way I do. Like I do the weirdest like I
Or
link.
we can say thank God it's food and food better. I feel like instead of thank God it's Friday, thank God it's food, and then food
Oh, I like it. Yeah, yeah.
better mucosal, I guess.
I actually like it. Okay, this is an amazing way. Keep that in mind, guys. Hala, moving forward, okay. We talked about the interaction between B and T cells. We said no, like let's go, like let's just really fly back. We said no, we wanna like form basically plasmablasts that release antibodies, and we said no, these antibodies they go through isotype switching and affinity maturation and somatic hypermutation, thankfully due to this interaction between CD40 and CD40L. Okay, now another thing that we want to create is memory B cells, and from the name memory B cells, they're B cells that just exist there to remember. So they'll forever remember this. Like, think of it like someone who's just resenting the body, like, like someone who's holding into the idea of this antigen and is really pissed at this antigen and is just gonna live forever waiting for this antigen to show up. Tamam? So this is what memory B cells are. So basically, memory B cells, they are usually produced in the germinal centers. And what did we say? The germinal centers are basically the place where we have activated B cells. And we said that the germinal centers, because they're not part of, like we have the primary follicle with unactivated, but then we have the germinal centers, they're usually secondary lymphoid organs. So what is the benefit of these memory B cells? These memory B cells, as we said, they're going to just sit back and just wait forever for an antigen that they know to show up. Why? So they can directly go and kill it. Tamam? It's like it's sitting back and resenting the moment that it ever met this antigen, and it's just waiting for revenge to go and like kill it. Tamam? They survive for really long periods and they don't really need like another stimulation. خلاص, they're stimulated. They have, like, you can look at it here. So basically, this is the interaction CD40L with basically CD40, صار عندنا class switching, and we had the IgG. The memory B cells, you can see it's old, it's just waiting there. And it already has the receptor and the antibody that it needs to release to basically kill the antigen. تمام? Perfect. Now, do you guys have any questions regarding this? Because we kind of finished the first lecture, which is the T and B interactions. Do you guys have any questions before we move forward?
Yeah, like I just missed the first part of like the
It's nothing. In the first part, literally, I only said, we have two cells, B cells and T cells, but it's nothing. Don't worry. You didn't miss anything.
no, the germinal center and secondary lymphoid. That's what it's kind of confusing me, you know?
Oh, it's okay. For the germinal center, let's go really fast back. So basically, we just said, no, the lymph node is like like an airport. Everything has to go through it, lymph, blood, whatever. And then the T cells are gonna both B and T cells are gonna enter. Once they enter, the T cells are gonna stick in the paracortical region, and then the B cells are gonna go to the peripheries, which are the cortical region. In the cortical region, they will reside all together and form something called the primary follicle. This primary follicle specifically, okay, if it gets activated, it's going to form another, it's going to form, you see this thing that's blue?
Yeah.
It's going to form something called germinal center, and germinal from the name, growing. So that's why B cells are activated, they're multiplying, there's clonal expansion, there's all of these things, all right? So the germinal center is something that's secondary, like that happens after. So it's a secondary follicle, okay?
Yes.
Oh,
Thank you.
you're welcome. Anyways, guys, the rest, do you have any questions? Just type no if you don't, yes if you do. Okay, slay. Now we're going to talk about the other. We've been talking about B cells, B cells, B cells. Okay, by then. Now T cells. Now for T cells, we're going to, I just wanted to revise. This is taban pre-midterm, and it's just important. Like I personally think it's important. I don't know if it's actually important, like for your exam. But we know, I know the T cell, for activation of T cell, we're going to have something called co-stimulation. And what is the co-stimulation? It's basically, you see this is the naive T cell. You can look at it, it's like cute and like it like never suffered anything in life, all right? So this naive T cell is going to basically, it expresses the T cell receptor. Obviously it's a T cell, it's going to have a T cell receptor, and it's going to express something called CD28. Now if we want to activate the T cell, something called co-stimulation happens, which is, we need two signals to simultaneously happen to activate the T cell. And what are these two signals? Basically we have the MHC class, I think 1,
Two.
I'm not, 2.
Yeah. CD4 goes with CD2.
Okay.
CD1 goes with MH plus.
Exactly. So what happens is we need the co-stimulation. The T cell receptor is going to, like the CD4 and the T cell receptor, they're going to interact with MHC2 that has the antigen. Simultaneously, the CD28 has to bind to B7. بصير عندنا an interaction for the T cell to be activated. If those two things do not happen together, the T cell is not going to be activated. حلو؟ حلو.
ممكن تعيد بشكل سريع. Can you repeat يعني in a quick way please?
Yeah, sure. Hello. Basically, for T cells to be activated, there's something called co-stimulation. From the name, co-stimulation, we need to stimulate at the same time. So what happens is we're going to have two receptors at play that have, like on the T cell, that have to basically interact with two receptors on the antigen presenting cell. In this case, it's a macrophage. It can be anything. It could be the dendritic cell, whatever. But it's an antigen presenting cell. So on the naive T cell, the two receptors that are there is the T cell receptor, which makes sense, it's a T cell, and something called CD28. تمام؟ هدول الاثنين, they need to interact with two other receptors that are found on the antigen presenting cell, which is basically the T cell receptor is going to interact with MHC class 1 that is presenting the antigen. So it's kind of, think of it like, I know it's kind of processing the antigen. The T cell, the receptor الموجود in the T cell, it's going to kind of try to know what antigen it is. Like it's going to process the antigen through interaction between MHC class 2 and the T cell receptor. Hello? Now, after this, at the same time, not after, at the same time of this interaction happening between the T cell receptor and the MHC class 2 that has the antigen, another like reaction has to happen, which is between CD28 and B7. So CD28 and B7, they have to interact. If those two, like those two receptors and those two receptors interacted simultaneously at the same time, only then will the T cell basically undergo differentiation. It will be stimulated, yani. Do you get it?
Thank you so much.
To simultaneously happen. And it's a very important, guys, keep it always in your mind. I don't know if you're going to be tested about it. I'm not sure. Like, I don't know if it was there in your midterm, but if it wasn't, I think it's usually a high yield thing. So keep it in mind. All right. Now, we said the T cells are going to get activated. طب activated لشو? Like we're activating it to what? So we have multiple kinds of T cells. We have طبعا نحن we know انه عندنا كمان something like we have T helper cells, we have cytotoxic T cells, بس I'm not talking about this. Now I'm talking about T helper cells. Okay, so for T helper cells we have multiple helper cells. Okay, we have type 1, not type 1, T helper 1, T helper 2, T helper 17, T follicular helper. You remember this? This is the one that helps us activate the B cell, and we have T regulatory cells. تمام؟ طيب يلا crash course. I mean quick question. يلا. T helper 1, what does it produce? What kind of what cytokine? Did someone say anything? So T helper 1, interferon gamma. صح. So T helper 1, always remember, and it will release interferon gamma. And okay, from your pathology information, interferon gamma, is it inflammatory or anti-inflammatory?
Anti?
Yep, it is anti. It's pro. No, it's not. It is pro-inflammatory. It causes inflammation, guys.
صراحة when it comes to like these T helper cells secreting and what they secrete and what's the job, cytokines, and it's like that's what I'm struggling with.
Okay, so it's fine. We can do them now
Excuse me,
together.
Talah,
Yeah, is it
your
still
voice is lagging.
lagging?
To me, it's perfectly fine.
Oh. But listen, guys, if it's lagging, type in the high. Maybe it's a connection issue with the host.
For me, it's good too.
It's good. خلاص. Okay. تمام. تمام. Okay. Everything's good. You can hear me, guys, right? Yes, okay. تمام. هلا T helpers type 1. Usually it's there in inflammation, okay? So because it's there in inflammation, what will it secrete? Interferon gamma. And interferon gamma, how do we type it? When you write it or you type it, you write I, F, and gamma, right? Inflammation starts inflammation, I and F, interferon gamma. So think of it and no, it's pro-inflammatory. It causes inflammation. Okay? So T helper 1 releases interferon gamma. It's pro-inflammatory. It's basically is نحن عنا whatever thing that can cause inflammation in our body, T helper cell is just gonna like run there. Okay? Then we have T helper 2. Yalla, who can tell me what T helper 2 secretes? IgE. Okay, this is a smart answer. IgE. So exactly, what is related to IgE? We just talked about them, guys. The 3 things.
Interleukin 4, and 3, and 13. Wait.
5 and 13.
Yes, 5 and 13.
Okay, so T helper 2 will secrete interleukin 4, 5, and 13. Okay? Exactly. It has to do with mast cells, with allergies, IgE, and we're going to talk about all of this in hypersensitivity reaction type 1. Okay? Now, T helper 17, from the name, it's number 17. So it releases interleukin 17, the easiest out of all of these. Okay? T follicular helper cell, basically, I'm not sure if it secretes anything, but it basically it secretes, I think, the chemokines that attracts the B cell. But just know like T follicular helper cell is just there for helping the B cell to mature. Now, T regulatory cell. Does anyone know why T regulatory cell is important? From the name, regulatory. So basically, T regulatory cell is something that will try to keep the body under control. طيب, regulation. Now, it stops things. Exactly, it regulates things. So نحنا in our body, okay, every single day we go through different phases. And I mean like in our body, there's a homeostasis between inflammation and anti-inflammation in a way, okay? Like, you get me? Like, think of it that way. So the T regulatory cell is there to kind of maintain this, tamam? And how does it maintain this? It maintains this by preventing like T helper cell 1, 2, and 17 from existing. Because if you think of it, T helper 1 is an inflammation. T helper 2 is in allergies, mast cell, like it causes desensitization. So command, they cause inflammation. And T helper 17 as well is involved in all of these things. So T regulatory is an anti-inflammatory. It keeps everything under control. And this cell is important to keep like the normal homeostasis in our body. And another thing is that it basically expresses a specific receptor that is more common in it, or like yeah, it's very important in the T regulatory cells. Can someone tell me what receptor it is? Like, do you guys know? No? No idea? Okay, I'll tell you. You guys can hear me, right? My kun, I'm lagging.
Yes, we can hear you.
Okay. So this receptor is interleukin-2 receptor. It's just literally interleukin-2 receptor. A T regulatory cell, why does it express interleukin-2 receptor? I'll tell you why. Because, let's look together. The funny thing is, no, the T cell needs to survive, right? So what does it do? It secretes interleukin-2. That's basically, I think, a naive T cell, okay? So, or like the naive T cells, the undifferentiated T cells, they secrete interleukin-2, 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 interleukin-2. But why? Because interleukin-2 is what keeps them alive. Because if you see, it will secrete all of these things, and then all of these interleukins are going to go and bind to the T cell. And this binding to T cell is going to result in colonal expansion, or basically, so it's funny because it's like the cell is what keeps itself alive, like how like this cell is so resilient, and it doesn't like give in into anything. So it's like, okay, you know what, I'm not going to depend on anyone, I'm going to depend on myself, I'm going to secrete anything I need to live. It's like a strong independent woman. Okay, I don't need anyone's help. Khalas, I'm going to secrete my own interleukin that will keep me alive. Okay, which is interleukin-2. So keep it in mind, guys. Interleukin-2 is important for what? Colonal expansion of T cells. تمام? And طبعا this interleukin-2, where is it secreted from? The T cells themselves. Now this is like summary of everything that we talked about. So yalla, fast recap. T helper 1, it's stimulated by, it's funny because it's stimulated by interferon gamma and it releases interferon gamma. Anyway, so this is pro-inflammatory. It's going to activate the macrophages and it's going to like start the inflammation cascade. T helper 2, it's stimulated by interleukin-4. You guys can see like a you can see شو يسموها like a pattern. So it's kind of stimulated by and it produces the same thing. Like it kind of stimulates its own self. But I don't think this row is important. The most important thing is these guys. So T helper 2 cells, it will secrete interleukin-4, 5, and 13. It has to do with mast cells, IgE switch, helminths, guys. And when we talk about IgE, IgE, so eosinophils. In eosinophils, what they usually like attack helminths, okay? And طبعا IgE and mast cells, both of them together. Then we have T helper 17. T helper 17, it releases interleukin 17, and this is basically, it has to do with the inflammatory process, specifically in neutrophil activation and like stimulation. Tamam? Last but not least, we have the interleukin 2, sorry, not interleukin 2, the T regulatory cells. The T regulatory cells, as I said, they have a very specific receptor for interleukin 2. That's why they're stimulated by it. They have, like, it's just called interleukin 2 receptor. And something that was mentioned in your slides, why do you guys think interleukin 2, sorry, not interleukin 2, why do you guys think the T regulatory cells has those receptors? Like, why not other cells have these receptors? Come on, like, why is it the one that has these receptors the most? Do you guys have any idea? Okay, I'll take the silence as no. So the idea is, basically those T cells, they have kind of, it's something genetic, like it's literally something in the makeup of these cells, and it has to do with FOXP3. I don't know how important this is, but I don't know if you guys know this. but Dr. Garwin only like focuses mostly on whatever is bolded in his lecture. فأنا like when I was a second year, I used to only study the bolded. But I think the FOXP3 was bolded, so I think it's important to know. Why is T regulatory cells? Why does it have interleukin-2 receptors more than the rest? Because of the FOXP3 in its genetics. تمام? Think of it, come on, this way. FOXP3, like a fox. Fox is sneaky. So it's trying to be sneaky. Okay, so T regulatory cells is trying to be sneaky. So it kills everyone else and takes over. Okay? Next. Now let's talk about cytotoxic T cells. What do you guys know about cytotoxic T cells? Cytotoxic T cells, from the name, cytotoxic. So they're the T cells that go and kill something on spot. خلاص I have a problem with you. I'm just gonna kill you. I don't want anything, you know, like I just want to kill you. تمام? Basically, how is it usually activated? It's usually activated by something called cross-presentation. And cross-presentation is just a fancy word to, or it's not a fancy word, like it actually says what it's happening. So cross-presentation, basically we're going to have simultaneous activation of a naive T cell alongside with a cytotoxic T cell through MHC class 1 and MHC class 2. So if you look at here, we have a dendritic cell. This dendritic cell is expressing an antigen because what's the job of dendritic cells? Just literally antigen. It's often called an antigen presenting cell. So it just presents the antigen. This antigen is presented on basically MHC class 2 for the CD4 T cells and on an MHC class 1 for the cytotoxic T cells. And then the signals, so basically both of them are going to be activated at the same time, and that's how a cytotoxic T cell is activated. Yani just the idea is, no, cytotoxic T cell is activated through MHC class 1 simultaneously with a CD4 naive T cell. Why? I don't know. هيك ربنا خلقها صراحة. I really don't know why. بس إنه the cross presentation has to happen. Okay? Clear? I think it's easy, guys. Yeah, that's it. Next. Okay, now let the fun begin. So I said cytotoxic T cells, they don't care. خلاص, they identified danger. They're just gonna kill this person. Or not person, يعني said, okay? So look at this. It's fighting. It's ready to fight. Okay? Now, how does this fighting happens? It happens through this. So it's going to have not the fighting, but no, it's going to have what is it called? It's going to have a receptor. تمام? This receptor is going to bind on the cells. So it's like a receptor that's kind of looking out for whatever is not okay or not good. Okay, so what does it do? It's going to bind to all the receptors on the cells, and if it identifies something that's not okay or something that's weird, like this doesn't feel right. So if it identifies a cell that is sick, that is tumor, that's whatever, cancer cell, or like a different cell, or like a whatever, what will it do? It will fight. How will it fight? By releasing perforin and granzymes. And what are perforin and granzymes? They're enzymes that will start attacking and kind of causing your favorite, probably not your favorite. Okay, I don't know why I said that. Anyways, it's going to cause apoptosis. So apoptosis is going to happen. Hello. What does apoptosis mean? It's just death. So apoptosis is usually triggered by the granzymes and perforins, and we know that apoptosis happen through two ways, right? We have an intrinsic pathway and an extrinsic pathway. Now, the cytotoxic T cells are going to kind of stimulate both pathways. So the granzymes and the what is it called? The granzymes. I'm not gonna go into the details of the intrinsic pathway because that's patho. And you guys already took the exam midterm for it, but what I'm trying to say is, the granzymes and the perforins are going to activate the intrinsic pathway. So what happens is, in the intrinsic pathway, the main thing is mitochondria, cytochrome C. The cytochrome C is going to activate the caspases, and then these caspases are going to result in apoptosis, and this apoptosis is gonna literally we're going to have blebs and it's just gonna kill itself. Okay. Now, it doesn't end here because come on. The cytotoxic T cell can cause apoptosis through the extrinsic pathway. هلا I just want to remind you guys, intrinsic pathway is triggered by granzymes and perforans, the enzymes. These enzymes are going to go to the mitochondria. They're going to start to like pick holes into it, break it down. The cytochrome C is going to go crazy. It's going to activate the caspases and the whole pathway is going to start. The extrinsic pathway happens through the Fas ligand. Okay, so the cytotoxic T cell, you can see it right here. It expresses something called Fas ligand. Fas ligand is just literally a death receptor. Okay, sorry, it's not a death receptor. It's a ligand for a death receptor. Like you can see here, if you look in here. طبعا this is something else. But you can see how we have something on the cell itself called a death receptor. So you know, basically, we have programmed cell death, like it's part of, you know how like they say death is part of life. So for the cell, come on, death is part of life. Okay, so they have specific ways to die. One of them is just expressing death receptors for the extrinsic pathway to happen in case of apoptosis. So one of these receptors is the Fas receptor. Now this receptor, it has a ligand, okay? This ligand is found there on cytotoxic T cells. So cytotoxic T cells can either send the granzymes and perforans that can activate the intrinsic pathway, or it could just literally two receptors interact, the Fas receptor with the Fas ligand, and then it's like, okay, kill yourself, and it just literally kills itself. تمام? There's command the tumor necrosis factor receptor 1. It's mentioned in your, I think it was mentioned in the slides. It's just TNFR1. So it's another receptor that it can work on command. It's just, guys, it's just the idea. I know it's a death receptor. What does death receptor mean? It's something that it binds to and it kills itself. Can you repeat? Sure. It's just the idea, guys, and in extrinsic pathway, or like the cytotoxic T cell, what will it do? It will stimulate death of or apoptosis of the cell, the cell that is cancerous, infected, whatever, a sick cell. Okay, how will it do that? It will do that either by the intrinsic pathway in whatever. Okay, intrinsic pathway by basically releasing enzymes, granzymes and perforans that are going to activate basically the intrinsic pathway. So they're going to like, the enzymes are going to go attack the mitochondria. They're going to activate cytochrome C, and then the whole caspases and the apoptosis happens. Or it has the extrinsic pathway. Extrinsic pathway, extrinsic from the name, extrinsic, outside the cell. Outside the cell, what do we have? We have receptors. These receptors are there. They're called death receptors. So they're just receptors that are just there for whenever the time comes, a cell might like attach to it and like trigger the death process. Okay, so in this scenario, we have something called a Fas receptor, and on the cytotoxic T cell, we have something called Fas ligand. Fas ligand means it's just something that binds to the Fas receptor. So this Fas ligand will bind to the Fas receptor. It will stimulate death, okay, by a bunch of signals. You can see that caspases are affected. It's not my objective صراحة to explain the extrinsic pathway. Just know and know Fas ligand will bind to Fas receptor, which is a death receptor, stimulating death. All right? And then there's another death receptor called tumor necrosis factor receptor 1. I'm not sure if you guys need to know it, but most importantly is the Fas receptor. All right? Is this clear? Can I move on? For everyone. You guys have any questions?
All clear.
Okay, so now moving on, we have the natural killer cell. Now the natural killer cell, it's special. Why is it special? Because it does not have a T cell receptor. Like, okay, I know it's a kind of a T cell, but it doesn't express T cell receptor. It's just unique, you know, just wanted to be unique. So it just was like, you know what, you all express T cell receptor, I'm not like you. كمان نفس الشيء, it does not express CD3, CD4 or CD8. So it's just like it just wanted to be unique. It's just like, I'm not like the others, and خلاص. It's just like, you know what, you all express T cell receptor, you all express CD3, CD4, CD8. I'm not like you guys. I'm not going to express these. But on the other side, it expresses something which is CD16. CD16 is important. Guys, when you see CD16, first thought in your head is natural killer cell. Come on? why is CD16 important? Because I'll tell you why, like we're gonna come to it. Now, the natural killer cell, the main function of the natural killer cell, it's kind of similar to the cytotoxic T cell, but we said in our body, like we said, we are kind of in a homeostasis between inflammatory and anti-inflammatory state. So we're kind of like we have a little bit of inflammatory, we have a lot of anti-inflammatory. We're just in a way, we're in an equilibrium, all right? So it's normal for like our cells, and subhanallah, yani, our cells like undergo multiple of mutations every single day, and our body detect these mutations, and they either stop the mutation or they kill the cell that is mutated. And how is this done? It's done by the natural killer cell. So what will it do? This natural killer cells, you know that MHC1 is so basically each and every one of us have cells, and all of your cells, like your skin cells, your GI cells, all kinds of cells, they will express MHC1 receptors. Why will they express this? This is kind of like a saying, like, hello, I am you, you are me, you know? So basically it's just trying to tell the immune system that I am part of self, like I am this human being, all right? Now, this is what will happen. Natural killer cells is going to go and it's going to basically try to identify, is this MHC1 the person's MHC1? If it is, then khalas, I'm not going to do anything. So this is a healthy normal cell. But if it goes to a cell and it does not express MHC1, then we're like, that's weird, because it's supposed to basically to express MHC1. It's not telling, it's not kind of, you can think of it, guys, MHC1 is like a white flag, and you're like, guy, like I'm you, you're me, please don't attack me. Okay, so if we don't have MHC1, then it's gonna attack it. If the MHC1 is not similar to the MHC1 that is found all over the body, okay, no, it's not the white flag, let's say it's a red flag, then come on, the basically the natural killer cell is just gonna like pew, kill it. Okay, how does this killing happen? So it's like the cytotoxic T cells in two ways, actually in one way, which is it's going to release granzymes and perforins, and you guys can literally see it. Sorry, you can literally see it in here. It's going to like just throw enzymes at the cell, and it's just gonna like ask it to kill itself. All right. And another thing that it does is it has receptors on its surface. You see
So
these?
the perforons are basically the one that it's gonna puncture holes, and granzymes are the one that, just a small recap, granzymes are the one that are gonna activate the caspases and the like
Yeah,
the
perforins.
extrinsic. Well, perforins is the one that's gonna puncture holes, صح?
Yeah, from the name, perforins cause pores. Granzymes are enzymes. It's gonna activate enzymes. I told you, you don't really need to know the details. Just know, it activates intrinsic pathway. All right, but that's it. Okay.
Yeah.
Don't like waste time on like teeny tiny details. Anyways, another thing is, now in here we have multiple receptors on the what is it called, on the natural killer cell. Now we have receptors that are activatory, or like it activates the process of killing, and we have receptors that are inhibitory, and they inhibit the process of killing. Hello. Now, what is the activatory process? It's basically this one, NKG2D. بدكم تحفظوها, يعني I'm really sorry to break it to you guys, but you have to know it. NKG2D, like a D, okay? Oh, you can think of it this way, guys. NKG2D, D for die, so it's gonna kill it, okay? So that's something I just came up with, but it works, okay? So NKG2D, the D, if you see a D, then that's an activatory receptor. If it binds to anything, then خلاص, it's just telling the cell, please go kill yourself. تمام? Respectfully طبعا. And on the other hand, those two receptors.
So this NKG2D is basically the receptor that is basically gonna tell the cell, cytotoxic T cell to kill itself, or I mean natural killer cell to kill itself, or it's gonna basically
No,
activate the natural.
It's a receptor that is found on the natural killer cell that will stimulate the bad cell that's sick or whatever to kill itself, you know?
Okay, I can memorize it in a way, natural killing, to die, basically. That's the way you can memorize.
That's amazing. So, sorry, guys. So, natural killer, it's for the natural killer to die, to die. Okay. Say thank you. هلا, another thing is anything that's, guys, إذا أنتم ما بدكم تحفظوا, if you don't have the capacity to memorize more, anything that is not NKG2D is basically inhibitory. So it will say, don't kill, don't kill, don't kill. So you can think of it in that way. Those two, they're going to bind to MHC1 that is self, and it's going to identify and know, oh, this person is a self, don't kill. NKG2D is going to identify everything that's foreign, and it's going to tell the cell, kill yourself. Hello? Now this is basically just this is the photo that the doctor had, so I just wanted you guys to like go over it again. So this is the target, and this is the MHC1. Hello? Now, inhibitory receptors are going to bind to MHC1. Okay, this is what are the inhibitory receptors again? The inhibitory receptors are anything that's NKG2, that anything that is not NKG2D, which is basically NKG2A. A is basically, you can think of it A alive, D die. So A is for alive, and cure, I don't know, guys. I feel with it, cure. Okay. Basically, this is going to bind to the HLA, and it's going to give an inhibitory receptor. So it's going to say, don't kill me, don't kill me, don't kill me, don't die. Okay, so it's not gonna kill. It's not gonna, or it's not gonna tell the target cell to kill itself. Okay. On the other hand, if an activatory like stimulus was there, so basically the NKG2D binded to a receptor, this is going to cause lysis. Okay, it's going to, like, the NK cell is going to tell the target, it's either going to kill it itself or just going to tell the target, just kill yourself. Okay, now we have another thing that's command special for the natural killer cells, and you guys can see the CD16 in here. Remember when we said CD16 is important and it's only found in natural killer cell? But now we're going to tell you why. So this is called antibody-dependent cell-mediated cytotoxicity. And from the name, antibody-dependent. So it depends on antibodies. And if you can look at this really sad sick cell, okay, you can see that there's a lot of antibodies around it. So it's kind of like tagging the cell. It's like opsonization. It's kind of tagging the cell. Okay, it's saying that this cell is sick. Why is it doing that? It's doing that to basically call for natural killer cells. So you see, these are the antibodies, and these are like kind of marking or like take it. I know, like it's calling out the natural killer cell. Come, natural killer cell, we have a problem here, and then it will come. It will, the CD16, guys. CD16 equals natural killer cells. This CD16 will bind to the IgG or whatever antibody that's there, and then basically we're going to have death. Allah yarhamah. Okay? Is this clear? This is easy, guys, from the name, antibody dependent. So we're going to have antibodies that are going to tag like cells that are either like filled with bad things, or they're sick or whatever, and they're going to interact with CD16. That's the only receptor that's very specific for natural killer cell, and this is going to cause the cell to okay, kill itself, or like the natural killer cell will do its job of like killing it. Okay, we can see in here it will produce cytokines, blah blah blah. Who cares? It's just gonna kill it. Okay. Now this is just, come on, as I asked earlier, like this is what the doctor, like the photo that we have with the doctor. So antibodies, they'll bind to antigens on the surface of a target cell. So they know that this target cell is, it can be, guys, it cannot be مثلا as, it can also be like a target cell. It can be bacteria, it could be whatever, like any cell that needs to be killed. Okay. What happens is these antibodies, they're going to interact with the CD16. Once they interact with the CD16, you see, basically, we're going to have something called a lytic synapse. Lytic means lysis. So it's going to, like, you can see things that are being released. These are basically the granzymes and the perforins, and they're going to cause apoptosis of the cell. All right, now we're done with the cytotoxic and NK and T cells lecture. Is this clear, guys? I feel like it was clear. We went over it multiple times. Can I move on to the tumor immunology lecture? okay, I'll take the silence as a yes. تمام. هلا I remember أنا صراحة, I'll be honest with you guys, when I studied for my POD, I skipped the slide. I skipped it. I was like, what the hell is this? I just skipped it. And guess what? I opened the exam and he got me a picture of that. I don't even remember what the question was, but when I saw the picture and I'm like, I just remembered that I got that in the exam. I don't even remember what was the what was the question or what was the answer. But you guys probably have like access to like whatever we did. You see that. If it's not there, just know that we got it. I'm a 13th batch girly, okay? Anyways, what is this? This looks sophisticated and it looks, at least it looked like that for me, okay? Like in second year. But it's such a stupid slide, guys. It just says that, okay, I have a mice. Because why do I have a mice? Because all of the experiments that happen in lab, they usually happen on mice. It's kind of sad, but yeah. So when they were trying to like do some studies to kind of understand tumor, tumor immunology is a field that is so interesting to me, and that's one of the reasons why I took this like the whole crash course, because I love oncology and it's one of my interests. Anyways, tumor immunology is a field that is so vast. There is a lot of research still going on there because we still don't know a lot of things. Okay, so why am I saying all of this? Just so you guys know and know basically the docs, like there's a lot of things that are says like, oh, evidence, experiments, blah, blah, blah. That's why, because nothing yet is 100% proven. So what do they do in this experiment? What they did is they got tumor cells, okay? Before anything, they got like irradiated tumor cells and they injected it to a mouse, okay? And then they left the mouse وما بعرف شو صار فيه بالmouse. Then later on, what did they do? They got the same tumor and they injected it again into the same mouse. Oh, an important thing to know is when they did this, like the first step when they did it, the mouse did not get cancer. Like خلاص, it didn't get anything. Okay. So then what did they do? They re-injected the same tumor and they injected كمان something different. Okay. Now, if you look at the end result in here, basically there's no tumor, but in here we got a tumor. What they're trying to say is, no, if basically, when they tried this the first time, okay, the mouse did not have a reaction because the body was able to fight it off. So the body had specific antigens, had specific makeup that was able to fight this kind of tumor cell. But that doesn't mean every single type of tumor cell can be fought by our body. Like, the idea is just the whole thing is just that, guys, if it's the same thing and the body was able to get over it, then it's gonna be able to like kill it again. But hon, it's a different tumor, so it needed different antigens, so it kind of couldn't eliminate it because it's a different cell type. Okay, so الزبدة is إنه نحن tumor rejection antigens, they're specific for the tumor, so each tumor has specific antigens against it. Okay, which makes sense I don't know why they like to. المهم, immune surveillance. Immune surveillance sounds interesting. Okay. The idea of immune surveillance, from the name, immune surveillance. We're keeping our immune system under we're keeping not the immune system. The immune system is keeping the body under surveillance. And why does it do that? Because we want to make sure and know we don't develop cancer. How does it do that? By trying to keep the body always at a anti-inflammatory state. It tries to like مثل شفتوا كيف natural killer cells, it's going around to all of the cells trying to see if the MHC is self or non-self. So this is all part of immune surveillance, or part of our body protecting itself from any danger. Okay, now what was the evidence for immune surveillance, or where did they get the idea of immune surveillance? Like, how did they know that this is something that we actually have? Basically, they did an experiment. In this experiment, okay, first of all, the experiment in here, the experiment that we just talked about, is kind of like an evidence to immune surveillance. لأنه نحن our immune body is مثلا this mouse had cells or antigens that were specific for this tumor and it was able to kill it off. and protect the mouse from it, but it wasn't able to protect it from another kind because it didn't have the antigens for that kind. Okay, so this is kind of like an idea of why, like how our body protects us from these things by immune surveillance. Okay, now how did they kind of come into it? So basically, infusion of BCG, which I'm pretty sure you know what that is. It's a vaccine in patients in early stage resected bladder cancer, is standard therapy for eradication of remaining tumor cells. So the idea is, this is too like advanced. I don't even know why the doctor like mentioned that, but I think just to keep you in the loop. So basically, guys, when someone is diagnosed with bladder cancer, all right, and it's in the early stages, what do? Usually the first line of therapy is to resect the bladder. They remove the part, they resect, not the, like they resect the cancer in the bladder, and they kind of like patch it and everything. So what do after resecting part of the bladder? They usually infuse, okay, in the like OR, they infuse BCG. Okay, and what is BCG? It's basically TB vaccine. Now, till this moment, they don't know what the idea is. Like, why? Yeah, I don't know also how they discovered this. I don't know if it was a mistake, if it was on purpose. Like, I really don't understand how they discovered this, but it's something that's actually like, it's in news until now. What happens is انه هنا نحن طبعا after you resect something, guys, you're not sure if you 100% removed everything. Sometimes you might leave a bit of tumor cells behind because we're talking about something that is on a cellular level. So that's why they infuse BCG to kill whatever tumor cells that are left. What is the mechanism under it? No one knows, but they only know that basically this BCG stimulates the immune system to kind of attack the tumor cells. Okay? You guys don't need to know this. Just know that we have something called immune surveillance. Immune surveillance is basically our bodies, like our immune system, like keeping everything under vigilance, making sure nothing goes wrong to protect ourselves. Okay? Okay. Now, the okay, this immune surveillance, or like the no, it's not immune surveillance, my bad. Tumor progression or cancer initiation goes through a phase. Hello? This phase, أول شيء we have the cancer initiation. So أكيد cancer is not gonna just pop like, ta-da, I'm here. Like, it's not that. It's not like that. Cancer happens due to a lot. It's like accumulation of mutations in the genes. So we have mutations over mutation for over mutation over mutation over mutation. I'm pretty sure you guys took this in pathology. You know oncogenesis and how does it happen. So cancer cell happens, okay? So the repeated stress, the mutations are going to result in the formation of a cancer cell. Now what happens? The first thing that happens is elimination. So our body is going to identify this tumor cell, and they're like, no, I'm not going to allow this. I'm going to protect the body. And you can see CD8 T cells, natural killer cells, all of them are coming here, okay? And they're trying to kill the cell. But طبعا what happens is they might be able to eliminate it, but if the cancer, like the cancer is caused by a mutation in a cellular level, it's gonna keep on like coming back and coming back. So what happens is we're gonna have something called equilibrium. What does equilibrium mean? So أول شيء elimination. So sometimes you can completely eliminate it and you're not gonna have anything. But if the mutation persists, if the stress persists, the cancer cells are still they're going to expand and expand and expand. And by the way, tumor cells are extremely, like, they're not smart, but they are smart. Like, they don't have a brain to be smart. You get what I mean? They're sneaky, they're smart. They will keep on trying to find a way to kind of escape the immune system. And that's like the last step. So after the elimination, so you're either successful and خلاص you eliminated it, or maybe you eliminate part of it. Then you're going to reach a phase, is خلاص the cancer is persistent. It's going to reach a phase of equilibrium, which is called basically tumor dormancy. From the name, tumor dormancy is that we have like cells that are basically, they're cooking, okay? They're going to like, the cancer is going to like unleash, but it's just dormant. So basically equilibrium between the immune system and the cells. So basically the amount of tumor cells that are formed are kind of the same amount that is being killed. So it's at the equilibrium. Then suddenly, no one knows how, actually like it kind of makes sense. So basically the tumor cells, I told you, they're smart, they're sneaky, they're trying to find a way to kind of escape the immune system. So they're going to find a mutation that helps them escape or evade the immune system, and that's when the escape happens, and khalas, hell breaks loose, and we're going to have the escape. Okay, so this is how basically cancer happens, tamam, or like the different phases that cancer usually is in. And just one second, basically there's something called cancer immunoediting. And this is what I was talking about. Cancer immunoediting is basically this cancer cells being sneaky, they're being smart. They're trying to see, okay, the immune cells are coming. What will they do? Oh, they will identify my MHC1. Okay. Let me find a way where they can't identify it. Let me put a barrier between me. So they're going to kind of edit immune editing. They're going to have mutations that's giving them an advantage to evade the immune system. All right, and this is like the different like parts of the cancer, and I personally find it very 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 fascinating. um uh the equilibrium, how do we know that something called equilibrium exists? From something called post-transplant lymphoproliferative disorder. Now, what is post-transplant lymphoproliferative disorder? From the name. So basically, I have a patient who had cancer, or like no, mother cancer. Like maybe he had a problem in his in whatever organ, and he needed to remove that organ and have a transplant. So some people will have something called post-transplant. After putting a transplant, they're going to have something called lymphoproliferative disorder. What does lymphoproliferative disorder do? And usually lymph هون doctor used the EBV virus as an example, but here the idea of lymphoproliferative disorder is that the body is going to start like overproducing. It's from the name lymphoproliferative. Proliferative يعني proliferate. So a lot of lymphocytes are going to proliferate, and all of this is going to result in basically result in like the disorder. All right, and this disorder kind of provides an evidence to equilibrium. All you guys need to know is the equilibrium state is proven by post-transplant lymphoproliferative disorder. That's it. Now, associating between defects in immune compartments and types of tumor. هلا, knockout. I don't know if Dr. Garwin explained what knockout means. Knockout is a very fancy word like that is used in research lab where they basically get a mice, and what do? They knock out specifically, and they go and remove the genes that code for مثلا perforin, or that code for RAG and STAT. Okay, so what do? They're like, okay, let's see if I remove the gene or the whatever material that like kind of like stimulates the how to produce perforin, what will happen? So what they found is, I'm not sure if you guys need to know this. طبعا the phenotype, guys, ignore this. The doctor had it like blanked out, so don't look at that. But the knockout, so perforin, if we don't have perforin, basically they realized, when a patient has deficiency in perforin, or when the mice did not have perforin, there was an increase in lymphomas. Okay, why? It's probably like the way the body prevents lymphomas is through perforins. Now, RAG and STAT proteins, you guys, I think you know JAK-STAT protein. It's a tyrosine kinase protein. And RAG has to do with like cell cycle and stuff. So when they removed these proteins, they realized, oh my God, we have a lot of breast tumors, we have got epithelial tumors. So it's just that they were trying to like figure out like, oh, like if I remove certain things, what will happen? All right. I don't know if you guys need to know this, صراحة. I don't know. But yeah, it's interesting. Okay. Now, we said no, basically we have something called tumor cancer immunomodulating, or basically like they're immunomodulating themselves to try evade the immune system. And there was a couple of like examples. I'm just gonna go over them really fast because they're not that important. So one of the ways that the tumor can kind of evade the immune system is by low immunogenicity. So they don't express MHC, basically MHC. So basically, if they don't express MHC1, the immune system can like not detect it. Okay, so basically downregulation of antigen presentation. So they don't really like have a lot of MHC1 and MHC2, so the body can kind of unrecognize the cell, all right? Loss of tumor antigens. So basically, we remember how we said in the mice, you remember how here in the mice we said, you know, each tumor might have specific antigens that help us kill the cell. So if we don't have these antigens, the tumor specific antigens, if they're gone, if they're dead, if they're, I don't know, mutated, whatever, this might increase the chances of the tumor like cells from evading the immune system. We have tumor-induced immunosuppression. So this is, guys, very interesting. So tumor-induced immunosuppression means the tumor cells, they know مثلا our immune system is triggered by specific chemokines. Let's say, remember how we said مثلا interleukin-2 is important to keep the T regulatory cells alive? So it knows that. And what will it do? It will just prevent interleukin from, exactly, like it will prevent interleukin-2 from being produced. فلأنه interleukin-2 is not produced anymore, the T regulatory cells are not, they're not stimulated, and because they're not stimulated, the the cell that is there to prevent inflammation, that secretes the anti-inflammatory cytokines, which is interleukin-10 and TGF-beta, is gone. So what happens is basically the tumor cell is just there and it's enjoying the inflammation. So it's kind of is like, you know how it's like, it's kind of distracting the body from the main problem by causing inflammation. Yalla, inflammation. So the body is gonna like kind of go into like shock and like, oh, what shall I do? Like, shall I deal with the inflammation? What is causing the inflammation? You know? Another thing is basically tumor release of collagen forming a barrier. So this is, come on, extremely interesting. And when interesting, I mean, it's just, it's crazy. Like, I don't know. Anyways, so what happens is basically the tumor cells, they just protect themselves. And how do they protect themselves? By releasing collagen or by releasing whatever. So they kind of form a layer around it that is hard for the immune system to kind of evade into. And it's just going to grow and grow and grow and grow and grow. It causes, like, it literally just forms physical barriers where we're going to have extracellular matrix. We're going to have collagen, fibers, or whatever, you know? Just to prevent the immune systems from going in. Wait. So interleukin-2 decreases, leading to a decrease in T regulatory cells and thus increase. No, it's not going to cause an increase in interleukin-10. It's going to cause a decrease in interleukin-10. Because let's remember together, what does interleukin-10 and TGF-beta do? They basically prevent inflammation. فإذا نحن we increase them, then they're doing the job. But no, when T regulatory cells is gone, interleukin-10 and TGF-beta is gone, فبصير في عنا increased inflammation. تمام؟ يعني everything you said is correct except for interleukin-10, the increase. It's gonna cause decrease in interleukin-10. تمام؟ and TGF-beta. Can you repeat the two? Yeah, sure. I'll go through them fast, but like they don't, you don't really need to know the specific details. Just know and know. We have certain ways where the tumor cells evade the no, it's totally okay. Don't apologize. So there are certain ways where the body is trying to, not the body, the tumor cell is trying to evade from the immune system. And how is it done? For the doctor gave you some examples. One of the examples is down regulation of antigen presentation. Or in other words, what they're trying to do is, you know how MHC1 can tell you if I am part of the cell or I'm not? So they can easily just either like do an MHC1 that is close to our body or basically not have an MHC1 and it can hide away from the cell, okay? Or we can have something called loss of tumor antigens. So we said at the beginning with the mice that our body has antigens that can fight the tumor cells. So what happens is, sometimes if these tumor antigens are gone, then خلاص نحن كأنه we left الملعب like للtumor cell. يلا do whatever you want. You don't have antigens against you. تمام? Those are two like mechanisms. Another mechanism is immunosuppression. And as we mentioned, immunosuppression, so the tumor cell is smart. It knows that our body needs chemokines, cytokines, interleukins, whatever, to survive. So what will they do? They will affect the T regulatory cell. How will they affect the T regulatory? They can prevent interleukin-2 from being produced. So when we don't have interleukin-2, the T regulatory cell is not activated. And if the T regulatory cell is not activated, what happens? Basically, inflammation happens. Why? Because we said a T regulatory cell is one of the most important T cells for anti-inflammation. It prevents inflammation. How does it prevent inflammation? By secreting interleukin 10 and TGF-beta. تمام? Last but not least, طبعا guys, this is just examples, like في كتير أكتر, okay? حتى if you can see, like the images that I got, it has like A, B, C, D, it's like up to J, but I just removed whatever like applies in your lecture. Anyways, last one, TME immunosuppression is basically just they form طبعا guys, TME means tumor microenvironment. Tumor microenvironment is just the environment of the tumor. This tumor is smart. It knows إنه أنا if I am out there, then the immune cell can find me. So what does it do? It kind of builds a wall, or it builds like a structure around it to protect it. So if أنا مثلا I protected myself like this, the immune cell cannot come and like attack me. It has to go through all of the things that I did here, which are basically, it can cause like release of collagen, increase of the extracellular matrix, just to protect itself. تمام? Now, we mentioned tumor antigens a lot. صح? We said tumor antigens, tumor antigens, tumor antigens. But what are tumor antigens? Okay. Now we have two kinds of tumor antigens. We have something called tumor-associated transplantation antigens, and we have something called tumor-specific transplantation antigens. So basically, just normal tumor-associated antigens or tumor-specific antigens. Now, what is the difference? The difference is what causes these antigens to form. هلا, the tumor-specific, they're caused by neoantigens, or basically new structures that are introduced into our body, or they're caused by oncoviral antigens, which are basically oncoviral antigens are basically viruses that causes, you know how certain viruses can cause cancer, like EBV, what else? I don't know, guys, like HPV. You know how those viruses can cause cancer? That's how they do that, by basically tumor specific antigens, okay? Sorry, like by stimulating like by releasing something called oncoviral antigens, and if basically we don't have the tumor specific antigens for them, then too bad, you know. So let's start with tumor-associated transplantation antigens. Now, tumor-associated transplantation antigens are tumors that happen they're antigens, okay, that are formed due to, wait one second, okay. They're formed due to our body, like overexpression of self-antigens, or antigens that are in our body that are misplaced. يعني مثلا, for example, the melanoma-associated antigen. The melanoma-associated antigen, it's an antigen that usually is found where? In the testes. So our bodies, not us, yeah, not females, males, but the male body is usually expresses, or yeah, it expresses melanoma-associated antigens. طيب هدول melanoma-associated antigens, where are they? Specifically in the testes. But if these antigens leave the body, or are expressed anywhere else but the testes, then we know we have something wrong. We know basically this means we have like melanoma, or like we have like melanoma cells that are like malicious, that are evil, they're abnormal. تمام. Another example is differentiated antigens. Differentiated antigens is basically, we have an example is alpha fetoprotein in liver cancer. So guys, I want you to, okay, the antigens, okay, the antigens are some things that the tumor cell like expresses, like like if these are present in the body, that there's something that's wrong, okay. هلا alpha fetoprotein in liver cancer, this يعني مثلا إذا نحن عندنا a patient who has liver cancer, they showed up with liver cancer. So what did they do? They want to see if it's, let's say, whatever kind of liver cancer it is. So what do? Like they do like an assay to check if the alpha fetoprotein is increased or not. Okay. What causes this alpha fetoprotein to produce? Basically, differentiated antigens. So they differentiate in a way that's different than the usual. So reactivated genes during tumorigenesis. So we have genes that are usually silenced by our body to protect ourselves. So if these genes that we were silenced, they were kind of like put down, if they're reactivated, they can result in formation of the tumor-associated antigens. And an example to this is alpha fetoprotein. Okay. Last but not least, another kind of a tumor-associated antigen is overexpressed normal proteins. Now, in breast cancer, okay? or in ovarian or adenocarcinomas. Some cancers, they undergo hormonal therapy. I don't know if you guys know that, hormonal therapy, or they undergo immunotherapy. Why do they do that? Because they have overexpression of this protein, which is HER2-neo, which is a receptor tyrosine kinase. It's a receptor tyrosine kinase that is found all over our body. But in these cancers, no, I don't know what happens. A mutation happens and we have overexpression. طبعا these things على فكرة, having tumor associated antigens sometimes can help in the treatment, because مثلا in this case overexpressed normal proteins for breast cancer, how do we treat it? We can treat it by a monoclonal antibody, or like an antibody that we generate against this tyrosine kinase receptor that can kind of calm down the tumor. تمام? Is this clear?
Can you kindly repeat 1 and 2?
Yeah, sure. تمام. هلا, tumor associated antigens, they're basically antigens that are produced by like the body, the tumor itself in the body, it will produce antigens. حلو. عندنا examples to these. We have something called melanoma associated antigens. هلا, this melanoma associated antigen, this antigen, okay, is usually expressed in the testes. So males, if you get any male and you get their testes, you do like you assay it and you try to find what are the receptors or whatever that expresses, you're gonna find MAGE or melanoma associated antigen. تمام? But if this receptor or this antigen or this whatever is activated anywhere else other than the testes, then that's when you know there is something wrong. Okay? So this كمان أصلا, it's this antigen is usually abnormally activated in melanoma cells. So when you have a melanoma, you know guys what melanoma is. It's type of a cancer that has to do with melanocytes. What are melanocytes? They're basically the pigment, the cells in our skin that cause pigment. So if a patient has melanoma, if they took if they took the melanoma, if they biopsy it or whatever, they can find MAGE, and MAGE is going to be abnormally activated. Usually it's not active, because where did we say this is usually found? In the testes bus. Okay? So this is an example to a tumor associated antigen. Another example is when we have genes that are usually like blocked out. All right? These genes, if they get reactivated, like مثلا, you know how basically you took in first year biochemistry, how we silence genes, how we basically, if مثلا you methylate a gene, then it's silenced. If a gene is silenced, then خلاص, it's not going to be expressed. So in tumorigenesis, or the formation of a tumor, we have a lot of stress in our body. Like the body is undergoing a lot of stress, and because the body is undergoing a lot of stress, like this is what causes the tumor to form. So this stress, that's the assault, this is going to cause some genes that were silenced to reactivate. So it's going to be like, oh, you want me silent? I'm going to break my silence, and it's going to become reactivated, and it can result in the formation of basically a tumor associated antigens like alpha fetoprotein, where in the liver cancer. تمام? Is this clear, guys?
Yes, thank you.
تمام. واخر وحده, it's easy guys, it's just a protein that usually exists there. Like it's a protein, like receptor tyrosine kinase, guys, it's a receptor, and I think you know that Trackstat, all of these are receptors that are found in all cells. Sometimes they can just go crazy, they overexpress a normal receptor, and then it's going to cause cancer. And this one is, guys, specifically HER2-neo is important. Why? Can you do it at the end, because they're being really strict about the time. They're like, you have to take, like, you know, just I'll let you do it, okay? بس بالأخير. المهم, ال overexpressed normal proteins, guys, HER2-neo إجاني in my final. I remember this. I don't know what the question was as usual, but this was there. فالأشياء اللي highlighted بهالموضوع, please go over them. All right? I hope that يعني شو اسمه, I hope إنه it worked. تمام? I know you guys understood it. Next, we have something called tumor-specific antigens. So as we mentioned before, tumor-specific antigens, they are antigens that were formed specifically due to an assault. And they're very rare. Why? Because usually the immune surveillance eliminates these. So when we say tumor-specific antigens, think of viral-induced or carcinogen-induced tumors. You know guys what carcinogens are. مثلا smoking is مثلا the tobacco particles in smoke is carcinogenic. Carcinogenic is anything that can cause cancer. You know, they're things that can cause cancer. So viral induced, okay, so sometimes viruses, as we said, EBV, HPV, they can cause cancer, or carcinogen induced. So when you see these two, think of tumor specific antigens. An example to this is HPV 16. We know that a human papilloma virus can cause cervical cancer. طيب, how does it do that? طبعا it does that through the tumor specific antigens, okay, or these antigens are expressed and you know that it's caused by HPV 16, okay. Another thing is mutated proteins. So some mutated proteins like cyclins and RAS and melanomas, these can cause basically, like, these are an example of tumor specific antigens, okay. What else? TCR or B cell arrangement or B cell affinity maturation, gene editing can produce. So كمان إذا تذكروا guys how we said إنه نحنا our body is like how cancer cells, not our body, how cancer cells do something called immunoediting. So they can cause, basically, they can inflict, or sometimes a patient has a problem in his immune system. يعني lymphomas are basically problems in the lymph, right? So cancer of the lymph, which is cancer of B and T cells. So هدول cancers that originate from the immune cell itself is usually due to like problems in the genes that usually that either code for the T cell receptor or the B cell receptor, or code for like the affinity or like the anything that can affect their maturation and their growth. Okay. These are tumor-specific antigens. Okay, so all of this is an example of tumor-specific. So for tumor antigen, it's something that is found already in our body that either is overexpressed or it's not in its normal place, it's abnormally placed. يعني مثل MAGE is normally found in the testes, بس إنه if we found it إذا كان عندنا it's expressed in anywhere else, you can know إنه oh this might mean it's a melanoma. What is it called? شو كان في another kind? The alpha fetoprotein. So basically in liver cancer, like hepatocellular carcinoma of the liver, which is usually like caused by either alcohol or whatever. يعني this kind of liver cancer, it's known for increased levels of alpha fetoprotein. Guys, انتوا فهمانين شو antigens? Like, do you guys get what antigens are? Antigens are things that are produced by the tumor cell that can tell you that, oh, I'm here, you know? Do you get me?
So basically, these are antigens that are naturally found in the body.
They're not naturally found. No, wait. These are مثلا MAGE is naturally found in the like, okay, I understand what you mean. They are naturally found in the body. بس هدول antigens, if they undergo something that's like wrong, if they mutate, if they're overexpressed, or if the differentiation has something weird in it, that's when they're associated with a tumor cell.
Meanwhile, tumor specific is basically we are producing antigens specifically for the tumor, right?
Yeah, like in a way it is specific. Why? Because HPV 16 is gonna cause, like, it's a viral induced, like it will cause something very specific, okay? Carcinogen is gonna produce something very specific. Associated, it's something that is found in the body, but it underwent like mutation, it underwent something that causes it. So these are oncogenes that turn into proto-oncogenes. Yeah, in a way. In a way, in a way. And you can think of it, it's something that we already have in our body, and the tumor associated, guys. It's something that we already have in our body, and these things, something bad happened. They either were misplaced, and then they were supposed to be in the testes بس راحوا على السkin, so oh, that's probably melanoma, or they were reactivated, and then they were silenced genes that got reactivated, or they're overexpressed. They're things that are found in our body, but something about their normal, they're no longer normal, they're abnormal. How are they abnormal? It depends. Maybe if they're not in their مثل ما قلنا melanoma, it's not in their melanoma associated antigen, like MAGE is not in its specific place, which is the testes. Alpha fetoprotein is basically maybe it was silenced and then it got reactivated. HER2 neo, it's basically overexpressed. It's usually expressed, let's say it's usually 20% of the time present. In this case, it's 100% present. Yani overexpression. Okay, so tumor associated antigen are things that are found in our body, but something went wrong and they became abnormal. Tumor specific antigens, there's some things that are يعني they're very specific for the tumor, as in like there's something that's outside of their body, outside of the body. And you think of it مثل ما حكينا, viral induced, carcinogen induced. It's something like it's an assault, it's a stress from the outer atmosphere into our body. يعني viral induced, we got a viral infection that turned into like a cancer. Carcinogen induced, you like introduced those carcinogens like fast food, like the smoking, like, you get me? I think it's clear now, guys. صح? Please say yes.
Yes, it's perfectly clear.
Okay, alhamdulillah. Okay, هلا we're gonna go into the treatment. تمام. The treatment, we have a lot of different. طبعا أنا guys, I don't want you to be scared from this. Like, into whatever is there in the slides, you follow. بس أنا هاي صورة, like, just to make it, I like visual stuff. All right. So because I like visual stuff, okay. Because I like visual stuff, I'll show you. So we have many different kinds. We have something called adoptive immune cell therapy. Okay, wait. أول شيء خلينا نشوف وين السرقة. Okay, adoptive immune cell therapy. In adoptive immune cell therapy, what do? They basically take this patients, they take healthy patient, they get it, they take their immune cells and they start working on it. Okay, they try to improve the immune cells. But guys, I love this topic because I like personally like had like ongoing lab research on it. Like it's amazing, guys. It's so fascinating. Okay, so basically adoptive immune cell therapy, what do? بيكون عندنا a cancer patient. They take this cancer patient. They're like, okay, let's extract their immune cells. And you can see basically they extract his T cells. They extract their tumor antigens, if there was any tumor antigens. They extract dendritic cells. They extract all of these things. And then what do? They try to improve it. How do they improve it? مثلا in CAR T cell therapy. CAR T cell therapy, the next slide talks specifically about CAR T cell therapy, but in CAR T cell therapy, they just improve the T cells, all right? So they're able to fight the tumor cell. يعني they just provide the T cell with the weapons it needs to kill the tumor cell. And it's so interesting, guys. Immune checkpoint inhibitors, from the name, immune checkpoint inhibitors. You know how we have cell cycle? I don't know if you guys took it. I think you did. Well, I don't remember شو كان POT, بس إنه في عنا something called checkpoint inhibitors, where basically when there is a mutation that happens inside the cell, okay, when oncogenesis happens, what happens? We have checkpoints in our checkpoints in the cell cycle, or checkpoints on the cells themselves, okay, where we have interactions. So in immune checkpoint inhibitors, we have something that's very like famous, which is the anti-CTLA4 and the anti-PD1. Okay, the anti-PD1, it's basically tumor cells. Okay, what they do is they express something called PDL1. Okay, high PDL1, it's, oh my God, guys, it's like a receptor. They're like, they express it, and they're like, it helps them kind of blind the immune cells from noticing them. So basically, يعني think of it this way. Think of it, it's like a trick. يعني أنا like, you know how, guys, you know when you wanna like, you have like, you know how you want to lure in the tumor cells, and you're like, oh, I have this PDL1, and our T cells, they express something called PD1. Okay, this PD1 will interact with PDL1, and it will just prevent an immune reaction. It's just like a way to blind the cells, and like, tara, I'm not a bad thing. Okay, so we have therapy, which are basically the monoclonal antibodies, which are antibodies that we generate, like we do, like مو أنا يعني, بس you get me, like they're antibodies that they create in the lab that can basically bind to this, prevent the interaction of PD1 and PDL1 to prevent this tumor evasion. Okay, CTLA4 is something similar to that. تمام. We have something called tumor vaccine. طبعا tumor vaccine is very it's a field of يعني they're still studying it to it's still very new. تمام. فإنه it's basically a vaccine that can help prevent cancer in a way. I don't know the details of it صراحة, so I'm not gonna FT. And then this is the CAR T cell therapy. Let's talk about the CAR T cell therapy. The CAR T cell therapy, what do they do? We said, you know, they do leukapheresis, which is basically taking the blood cells. These T cells, they're purified. They reprogram the T cell and they give it the weapons it need, basically receptors, whatever. They give it any weapon they need to fight the cancer cells. After they incorporate this receptor or this weapon, they do expansion. They kind of like just cause like clonal expansion. You'll expand, and then they just inject it back into the body so it can fight the tumor cell. Right? Guys, I don't care about this صراحة خلاص. You guys can read this on your own. Like, you know, fast ligand, fast causes apoptosis. These cause down regulation. You remember guys, عم قلكن أنا PD مع PDL1 can cause these like that can cause the tumor cell to basically evade the immune system. So it down regulates the fighting. Okay? That's the right word. تمام? Guys, it's clear.
The
It's fine.
last treatment, which is CAR T cell.
Eh.
Can you please يعني
Yeah, sure. I can re-explain it. It's okay.
thank you.
Can I
So the
re-explain
idea
it so that I make sure I understand it too?
Yeah, sure.
وانت you correct me, okay?
يلا.
So basically, we're taking the white blood cells, which are found in the bone marrow, and what we're gonna do is that we're gonna take the white blood cells and we're gonna try to proliferate them. We're gonna try to reprogram them so that they attack only the tumor that we want. So we're
Yeah.
gonna try to change the antibodies or the antigens that they have.
We don't change anything. بالعكس, we give them the weapons they need. يعني we change their DNA in a way that gives them an advantage against the tumor.
Yeah, that's what I meant.
Yeah.
And after we reprogram them, we're gonna proliferate them so that we have more. Basically, we're taking the tumor and you're making it more so that it can attack the tumor.
You mean the cells?
The cells, the tumors. Yeah, the T cells, I mean.
Yeah. And then you reinfect them.
That's it. That's it.
طيب, is it clear, guys?
Yes, thank you.
أكيد? Okay. يلا, let's move on. Okay, خلصنا هلا immunogenesis. هلا خلصنا tumor immunology. Now we're going to hypersensitivity. And guys, I'm so excited for this because I got you the cutest. Look, it's so funny. Let's see.
But can we like have a 5-minute break عشان we don't burn ourselves up?
The issue is they're being so strict about the time. And she wants me to finish, like she wanted me to finish in 2 hours, which is impossible. I told her that. But I don't know. I'll go slowly, okay? Is it okay?
See, the thing is like that's the thing, like they want to finish it early, but أنا بحس we're being over rushed. وما عم يعني I like to like take my time and I don't like someone being rushed, you know?
Yeah, I get you. But I can't do anything about it.
Yeah, I get you.
So let's go into hypersensitivity reactions, guys. This is so exciting, okay? So for hypersensitivity reaction, let's start with hypersensitivity reaction 1, okay? Hypersensitivity reaction type 1 is basically the immediate hypersensitivity reaction. Hello? And by immediate, we mean an assault happens and directly we have a reaction. So another name for hypersensitivity reaction type 1, you can think of allergies, anaphylaxis, all of these things. So what happens is you see this poor guy, he's out there smelling the flowers, and then an allergen goes up to his nose. This allergen is going to go into the dendritic cell. This dendritic cell looks at the allergen, is like, it's foreign, it looks obnoxious, repulsive, I don't like it. So it runs, you see how it runs to the naive T cell, and it's like, look. And then the naive T cell is like, oh my God, this is a foreign cell. Oh my God. So this is when the T helper cell type what, guys? Which one? هي مكتوبة بس إنه which one?
Type 2, which is gonna secrete interleukin 13, 4, and 5, right?
Exactly. So you see, T cell, T helper cell 2 is like, oh my God, it must be a war. Yalla, let me go and wake up B cells. Okay. So it goes like this. This is T helper cell. It will secrete IgE. It will secrete interleukin. Sorry, no. It will
Interleukin
secrete,
4 or 5, which
exactly.
is
It will
gonna activate IgE, which is
yeah, exactly.
from the
The
B cells, right?
Yeah, exactly. So what happens is the the T helper 2 type cell is going to secrete interleukin 4, 5, and 13. This is going to go to the B cell, and remember we said in a certain cytokines as stimulate production of specific antibodies. So the B cell is like, okay, I'm going to produce IgE. تمام? So it will produce IgE. But it's not gonna produce it this يعني شوفوا, it's gonna produce IgE. هلا this IgE that's produced by the plasma cell is gonna go to the mast cell, and then it's going to cause the mast cell to feel overwhelmed. She's the mast cell is like looking at all of these IgEs and it's like, oh my God, I feel overwhelmed. Okay. So it feels overwhelmed and it feels sensitized. Okay. But guys, keep in mind, first exposure, nothing happens. يعني أول ما أنا am exposed to an allergen, okay, all of these happen, but nothing, no symptoms are found. Okay. So in the first exposure, all of this happens to sensitize the mast cell. Okay. So the sensitized mast cell, in a way, is kind of like the memory B cell. تذكروا the memory B cell where I said إنه it's like it resents the antigen, and it's like, oh, where is this antigen? If I see it, I'm gonna like kill it and send all of these. So the mast cell is kind of like the memory B cell. It's sensitized. Now, when this guy goes again to the garden, okay, and he smells again, and the allergen comes back. So look, the allergen is like, oh, hi, I'm here. So the sensitized mast cell, I think it's holding into the idea of this allergen, and if I see this allergen, I'm gonna kill it. So the mast cell is gonna like recognize the allergen, and then it's going to it's going to send all of, it's gonna like degranulate, it's gonna send all of these granules that will result in basically vasoactive amines to be produced, cytokines to happen, lipid mediators, and this is going to cause the vasodilation that we see. It can cause the bronchospasm. You know, when we have an anaphylactic shock, not shock, sorry, anaphylaxis, my bad, it's not guys, don't, not anaphylactic shock, okay, anaphylaxis, when anaphylaxis happen due to an allergic reaction, so they're the first thing they'll be like, I can't breathe, I can feel my like epiglottis closing. Why? Because we have bronchospasm and we have edema, or like edema that's caused by the vasodilation and increased permeability. So the star of this short, the star of hypersensitivity type 1 is the mast cell. Okay, the mast cell. Thank you guys. Well, I made my day. Anyways, the what is it called? Okay, so mast cells, they're the like, they're the ones that are like, they're the boss of the show. So these are some like very like, these are the things that we already talked about. You know, they have high affinity for IgE, so IgE will bind to them, and after it binds to them, what happens is we're going to have cross-linking. Yani, guys, this is things that you know, I'm not gonna go over it to the time. Okay, this is what we talk about, like when we say it's an immediate reaction. So like after the secondary allergen exposure, we're going to have the cytokines are going to be released, degranulation, yalla, a lot of things are going to be released, and we have another part of it, which is the late phase reaction. Now for the late phase reaction, usually TNF alpha is released, tamam, by mast cells. Okay, that's all you need to know. You need to know basically it's type 1, it's predominantly the cell that is predominantly related to it as mast cell, IgE, blah, 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 blah. Okay, we have a late phase of this reaction where TNF alpha acts. وطبعا guys, if you think of it, it makes sense. Why? Because if you look in here, when we increase vasodilation, when we cause all of these things, when we cause cytokines, بيصير في عندنا inflammation. And what is the most important mediator in inflammation? TNF alpha and interferon gamma. تمام؟ Okay, now in here we're just talking about what are some examples of type 1 hypersensitivity. We have hay fever, food allergies, anaphylaxis, all of these are examples. How do we treat type 1 hypersensitivity? Think of it. Basically, the problem is in mast cells. The problem is in IgE. The problem is in T helper cells. Okay, so what do? We inhibit the mast cell degranulation by a medication called cromolyn. Cromolyn is basically it inhibits mast cell degranulation. So you see this step where the mast cell is going crazy and it's just releasing everything. So cromolyn is gonna come, it will bind to this mast cell and it's like, mast cell, relax, no need to overreact, it's fine, don't do anything. So that's what cromolins do. Okay. في عنا شيء اسمه antagonize effect of cell mediators. So basically نحن we know انه histamine is gonna be released, like نحن we said, okay, this is something we did not say, but we said إنه عنا هون leukotrienes and arachidonic metabolites. So histamine is kind of a type of these vasoactive amines, okay? And of these, like, hey, histamine is going to be released because into أصلاً, you know, guys, like if you have allergies, if anyone in your house has allergies, they're going to take antihistamines. So that's what they're trying to do. They're trying to calm down the mast cells. They're trying to reduce the inflammation. So they're going to give them corticosteroids, okay? Specifically in anaphylaxis. يعني into أصلاً, you know, guys, if someone is going through anaphylaxis, what do? EpiPen. So epinephrine is there to reduce the inflammation to allow the patient to be able to breathe. And desensitization, طبعا this is, guys, more than your level, just ignore it. Just know that there's something called desensitization. I can explain it really fast, but you're not supposed to know it يعني in details. Desensitization is just from the name, you're trying to desensitize the patient to the trigger. So if I am triggered by a specific allergen, I keep exposing the patient to it, exposing لحتى يتمسح، لحتى خلصك. I don't care anymore, you know, like, oh, like, let it come, I'm not gonna react. Okay, that's it. Now type 2. But guys, قبل ما نروح على type 2, I want you to keep in mind type 1 allergen, type 1 immediate hypersensitivity type of reaction, IgE mediated. احفظوها, like, I don't know, like IgE mediated. احفروها بمخكم. Okay.
You can remember it as IgE, E for egg, and egg is an allergy. So type 1.
Okay, that's actually interesting. You can, guys, keep that in mind. Now let's move on to type 2 hypersensitivity. Now, type 2 hypersensitivity, okay, it can get tricky to differentiate between type 2 and type 3. فركزوا معي, تمام? Type 2 hypersensitivity is antibody mediated. يعني it has, like, it's predominantly caused by antibodies, and not any antibodies. It's specifically caused by IgG and IgM. Because if you guys think of مثلا type 1 immediate, we have IgE, فإنه IgE is an antibody كمان, صح? But no, in type 2, it's antibody mediated, specifically IgG and IgM. Okay? And it kind of يعني ما بعرف هلا. Okay, this is just like, guys, this is we're like warming you up. So what happens is this is an what is it called? This is an antibody. So it looks at this poor cell and it's like, don't keep these antigens, like خلاص, like just get rid of these antigens, okay? And then it's like, but these antigens are part of me, okay? Like, those antigens are my antigens, like they're self antigens. Can you not see, they're self antigens? And then the antibody will react and was like, no, I will kill you, and it attacks it. Okay. طبعا هلا انتوا هون, you guys are thinking انه مو نحن حكينا انه like self antigens, we don't attack our body, like our body does not attack itself, right? هلا in type 2 hypersensitivity, there is a problem, like there's an underlying problem that causes the antibodies to attack whatever kind of cells. All right, so let's start. هلا, this type 2 hypersensitivity reaction can be caused by so many different things, okay? And the mechanism of action is important. هلا, it can be caused by drugs, okay? So sometimes if you take a specific drug, it can cause the body, it can cause the body to go crazy, and the IgG and the IgM are going to go and attack anything. They're just going to go and attack the body. You can see in here, like the antibodies, they're attacking an antigen, and then they're just causing opsonization and phagocytosis. They're just like, oh my god, they're overactive. Why? They're caused by these who are basically drug reactions. Maybe a drug can increase the amount of IgG in our body. Maybe transfusion reaction. When we say transfusion reaction, what do we mean by transfusion reaction? Transfusion reaction means I am يعني لا سمح الله in the ER. I need blood to be transfused to me. They did not they did not like properly cleanse the blood. Or يعني مثلا هلا basically guys, when you transfuse blood, what happens is before they transfuse any blood to another, they kind of go it like they could make it go through different phases where they filter the blood. They make sure انه بالبلد ما في antibodies or ما في antigens that can trigger my body. Sometimes they don't do that perfectly, or sometimes there are things that we can't just remove, for our body are going to find them and they're going to start attacking. You get me? But type 2, it's IgG and IgM mediated, and it can be caused أول mechanism of action either by a specific drug, or it can be autoimmune or transfusion reaction. Okay? Now, let's go into more like details of more examples. هلا عنا specific diseases, okay, where they're known as type 2 hypersensitivity reaction. هلا guys, you know, you don't know that. Never mind. Okay. طيب هلا نحن عنا specific, or you guys should know this. Wait, you know rheumatic fever? You should know this from micro. Or do you know?
Yes,
Okay.
we heard it.
Okay, perfect. Okay. So what happens, we have something called هلا هون حاطين لنا in this thing, they showed you the 3Gs. The 3Gs of the inflammation. Okay. So what happens is you can see these are basically basically this is the antibody. That is the star of the show. اللي هي الantibody that goes and it binds to the antigen. بس هيك for fun, like, you know, like, oh, IgGs and IgMs are just bored and they're like, oh my god, let's go and like attack random cells. Okay. هلا why do they attack random cells? Like what is the cause of this? So it can be due to like مثل rheumatic fever. Now rheumatic fever is caused by, what is rheumatic fever? So we have, you guys know gas, which is basically streptococcus pyogenes. Streptococcus pyogenes, all right? It's a bacteria. Now, this streptococcus pyogenes or strep pyogenes, it causes mainly pharyngitis, or it causes like respiratory problems. Now, if you do not treat this in the best way, and if you don't directly treat it in the way that you should treat it, what happens is بصير something called molecular mimicry. And by molecular mimicry, what does that mean? Our body is going to mistake our own cells to whatever it attacked due to this bacteria. By molecular mimicry, we mean مثلا عنا this strep pyogenes. I don't know if you guys took this, but it has a virulence factor which is called the M protein. Do you know that? Did you take this?
Yes, we did.
Okay, slay. هلا هاي الام protein, the structure of the M protein, ما بعرف كيف, it's kind of like the structure of our heart. Okay, in a way. What happens is إنه our body, when it's trying to fight off this bacteria, it will create antibodies against M protein. Okay, هدول antibodies, they can mistake our heart cells إنه يعني they can go and they will think إنه our heart cells are those M proteins, and they'll attack it and they will cause basically rheumatic fever. هلا glomerular nephritis, same thing happens. What happens is إنه بصير عنا molecular mimicry, where something that's released by a bacteria, like a virulence factor or whatever, our antibodies يعني think إنه a structure in our body is similar to that thing and will start attacking it. So in glomerular nephritis كمان على فكرة, which is part of strep pyogenes, بصير في عنا the body will start attacking the kidneys. Like the antibodies are just gonna go and they're going to, مثل ما شفتوا كيف هون, the antibodies are sticking into these cells. Now, because they're sticking into these cells, شايفين هدول الcomplement? You guys know the complement cascade, C3 blah blah blah, C3 blah, blah, blah. You know these? Okay. So one of the ways where our antibodies like work نحن بنعرف إنه antibodies how do they work? They work either by opsonization and phagocytosis, so they either like mark a cell and they're like calling the macrophages يلا come eat the cell, يلا come phagocytosis and kill it, or they call the complement, they activate the complement cascade and all the complement cells are gonna come. So in type 2 hypersensitivity, all of these happen because the idea is the antibody is attacking the body itself. Okay, so glomerulonephritis, Goodpasture syndrome, all of these are examples of type 2 hypersensitivity where our body is attacking itself. So the antibodies are just going and they think they know the cell, they think the cell is يعني this is what molecular mimicry means. أصلا أنتم من الاسم molecular mimicry. So they think the heart cells or the kidney cells or all of these things, they're mimicking the M protein or whatever virulence factor, and they think ما شاء الله عليهم they're so smart, and they go and they attack them. They think هنا مفكرين حالهم عاملين شغلة منيحة يعني. They think oh my god I'm protecting the body, but they're doing the opposite. All right, so this is type 2 hypersensitivity. Last but not least, another example, we said guys, antibodies act on the body by opsonization and phagocytosis. They act by basically activating the complement cascade. And last but not least, they do antibody-mediated cellular dysfunction. And what do we mean by antibody-mediated cellular dysfunction? So you guys, I don't know if you know these diseases, but they were mentioned in the lecture. I don't know how the doctor explained them. But هلا عندنا two diseases that are called Graves' disease. Graves, guys, you took in the repro. Perfect. طيب, what is the problem in Graves' disease? We have can we recap type? At the end, I'll recap everything in the type 2. بس خلونا نكمل. This is type 2, by the way. We're still in type 2. Graves' disease. You guys remember Graves' disease? Graves' disease is basically, it causes hyperthyroidism. How does it cause hyperthyroidism? Our body, ما بعرف هيك, our body goes crazy and it starts producing antibodies that mimic TSH. So this TSH, so you see this antibody is going to go to the TSH receptor and it's going to tell that it will, so you see here, شوفوا أصلا شو هون كاتبين, now you're hypnotized, you'll do what I say. So this antibody, okay, that is gonna bind to the TSH receptor. So when an antibody binds to the TSH receptor, what happens is, like normally, when when it binds to the TSH receptor, it's gonna activate the TSH receptor. But in this case, this antibody, this random antibody like IgG or IgM, is gonna go and bind to the TSH receptor, and it's gonna hypnotize. It's gonna be like, look, I am whatever. Okay, wait, agonist and then MG is antagonist. Yeah, exactly. So it's gonna kind of gaslight it and it's gonna hypnotize it, and you're like, hello, I am a TSH. Okay, so yalla, start secreting. So it will agonize TSH receptor. So the thyroid hormone, the thyroid gland is just gonna go crazy and it's just gonna produce thyroid hormones, and that's how hyperthyroidism happens. Okay, is this clear? Guys, I'm just giving you examples that are inside the lecture. Like أنا ما زودت أي شيء. The doctor mentioned these. I don't know how he will ask you, but I want you to understand the concept. Type 2 hypersensitivity is due to IgG, IgM. Those IgG and IgM, they either can just ما بعرف go crazy and they'll just go and opsonize random cells, okay, and cause the macrophages to eat them up. And this is usually due to like drugs, drugs you take, transfusion reaction, whatever, okay, or it's due to inflammation اللي هي molecular mimicry. So the body thinks, oh my god, I'm saving the world, I'm saving the body, and it thinks مثلا the heart cell or the kidney or whatever, it thinks it's a bad guy, and it goes and it activates the complement system, which will cause basically all of the symptoms that happen, okay? Or it can act like an agonist. It can gaslight something into thinking, oh, by the way, I am the receptor, like I am this hormone, yalla, do my work, just like in Graves' disease.
So in the mimicry part, basically the antibodies are gonna go and like not attack بس يعني bind to the normal cells, and then that's gonna lead to an inflammation or like the
Yeah, it will
clear-
lead to an inflammation. Why? Because it will activate the complement system.
Okay, thank you.
Now let's explain Myasthenia gravis. Myasthenia gravis is the opposite of, it's the opposite of Graves. It will antagonize the receptor. So شايفين these are the antibodies. Okay, and this is the neuron. So you know, guys, I don't know if you know what Myasthenia gravis is. Myasthenia gravis, you're gonna take it in MSI. Did you guys take it in endocrine? I'm not sure. Anyways, Myasthenia gravis, the idea of Myasthenia gravis is they're going to have weakness in their muscles. We took it in MSK. Okay, slay. So you know what Myasthenia gravis, we have weakness in the muscles. Why do we have weakness in the muscles? Because they're not being excited. Hala, you see this neuron, it's excited, as I see, it's smiling, and yalla, let's release acetylcholine. So it releases acetylcholine. But if you look at the antibody, okay, it blocks the receptor. شايفين ايديها كيف عاملة هيك? It's blocking the receptor. So the acetylcholine cannot bind. So what does it do? It's telling it, don't you dare to attach. I blocked it. Like خلاص. Okay? So it's gonna prevent the activation of the muscle. So we're going to have Myasthenia gravis. تمام? So it's an antagonizing effect. So in Graves' disease, it's an agonist. In Myasthenia gravis, is an antagonist. حلو? Is it clear, guys? Just know the basic, which is type 2 hypersensitivity reaction equals IgG, IgM. These IgG and IgM, they're going crazy. Why are they going crazy? We said why. It's either because of drugs and transfusion reaction, or it's because of molecular mimicry, or it's because of طبعا molecular mimicry is caused مثلا by an infection, مثل strep pyogenes, whatever, or I don't know, it's just decided to go and like gaslight the receptors into thinking they're either working or not working. تمام? Is it clear? You don't need to know every single detail. المهم تكونوا فهمتوا the concept. إذا أنتوا فهمتوا the concept, that's all you need to know. خلاص. الحمد لله. Guys, I'll confess to you something. إنه I struggled with hypersensitivity reactions up until now, my 5th year. Like I did my step exam like recently, so when I did it, like before I do it, I was like, I still struggle with hypersensitivity. So I hope يا رب إنه أنا أكون اليوم ساعدتكم and you forever just know what hypersensitivity is, because I really struggled. هلا خلصنا من type 1 و type 2. Let's go to type 3. Okay? Type 3 is okay. هلا نحن حكينا إنه type 1 and type 2 can be kind of type 2 and type 3 can be kind of misleading. Why? لأنه type 3 كمان has to do with IgG. بس it doesn't have to do only with IgG. It has to do with the idea of IgG binding to something, causing the formation of a complex. So when an antibody binds to an antigen, it's going to cause an antibody-antigen complex. Now, the problem in type 3 hypersensitivity, we have a lot of antibody and antigen, like a lot of immune complexes forming randomly for no reason. They're just forming. And these immune complexes, what will they do? They will go and they will be deposited in various parts of the body. They can be deposited in the muscles and the liver. They're just going in the blood and they're just falling anywhere. Whenever they fall, okay, they're going to cause basically complements to be activated. So شايفين هون C3b and C5a, they're going to find this complex and they'll be activated. Once they're activated, these طبعا C3a, C4a, C5a, they're called anaphylotoxins. So what does anaphylotoxins mean? على فكرة guys, from the name, anaphylotoxins, we said anaphylaxis يعني عندنا mast cell degranulation. فهون بصير في عندنا mast cell degranulation وبصير في عندنا opsonization by C3b. So it will cause an immune reaction to happen. Okay? And the whole, like the different kind of whatever, يعني مثلا if it's affecting the kidneys, we're going to have kidney symptoms. If it's affecting whatever, it's going to have whatever, you know? So, do you understand it, guys? The difference between type 3 and type 2 is that in type 2, it's the antibodies binding to whatever cell and causing damage. Hon, it's the complex of the antibody with the antigen depositing in different areas, activating the immune system. Do you get me? Please say yes. Yeah? It's easy, guys. It's at the complex. الcomplex هو المشكلة. The antigen مع الantibody depositing in different places is what's causing the problem. تمام? Please say yes. Just type yes, please. Can you hear me?
Yes, it's clear.
Okay. تمام. So Arthas, and doctor had this Arthas reaction. You see, guys, إنه basically عنا ما بعرف عنا immune cells مع antigens. They're forming complexes. These complexes, they're just here. They're just deposited in the skin. And then this deposition causes inflammation. And you can see here, guys, كيف like it's red because of the inflammation. I don't care. المهم هون هلا the treatment, you guys don't really need to know the treatment because the doctor did not have it bolded, but for type 1 you need to know it because it was bolded, okay, and it makes sense. هلا هون you just want to limit the inflammation. You really can't do anything. يعني عن جد, guys, you can't, like, if the antibodies are going crazy, what can you do? There are ways to prevent them, but كمان they're very expensive. So أول شيء, you try to limit the inflammation. How do you limit inflammation? You try to suppress the immune system by giving corticosteroids. So you're telling the immune system خلاص انطم and sit down. You know, don't do anything. هلا إذا كان ال type 2 and type 3 very severe, we do something called plasmapheresis, where basically they it's like dialysis of the kidney. إنه كيف الurine البلد كله بيروح, they remove the toxins. The same thing. The blood is going to go into a machine and it's going to remove all of the antigens. Okay? We can try to basically deplete it. We can try to block the antibodies by monoclonal antibodies. بس خلاص يعني, guys, you know, like, we're just gonna try to stop the immune system. Okay? Now let's go into the last what is it called? Hypersensitivity reaction, which is type 4. هلا type 4 is different and it's delayed. Guys, it's funny. Think of it that way. Type 1 is immediate, type 4 is delayed, and then in between it just has to do with antibodies. You know? بس انتوا أكيد تعرفوا what's the difference between 2 and 3? 2 is antibody attacking cells. 3, it's the antibody antigen complex being deposited in places causing inflammation. Okay? Okay. Now, هلا delayed hypersensitivity type 4, it's basically cell mediated. And when we say cell mediated, we mean we have T cells. So these T cells are the cause of inflammation, because if you guys think of all of the previous ones, T cells were not involved. يعني the T cells, they were not directly involved. It was always the B cells releasing antibodies. Okay, type 4 is cell mediated, so the T cell is going to cause inflammation. وطبعا it's delayed. Why do we call it delayed? Because it happens like 24 to 48 hours after the assault. Because it's cell mediated, so the inflammation is going to happen it's going to be caused by the T helper cell 1. So guys, شو قلنا T helper cell produces? It produces interferon gamma. Why? Because a T helper cell causes inflammation. IFN F gamma. Okay. بعدين عنا T helper 17 كمان will release interleukin 17. So everything that can cause inflammation is going to be released. تمام? That's all you need to know about here, صراحة. Do you guys know what test is this? Okay. Mantos, TB, exactly. So the idea of this test is that tuberculin test, skin test, right? The Mantos test. So the idea is, you know, they know that we have something called delayed type, like so basically, to test for to TB, they kind of use the concept of like, oh, I know, basically our body produces immune, like, the cell mediated, like, they know, type 4 exists. So what do? They inject a little bit of inactivated TB into the skin, and they wait to see, will it swell or not? Because if it swells, this means يعني our body knows how to act against TB. So this means our body is sensitized to TB. But إذا ما كان في عنا an increase in size, so this means the patient never had TB, so ما كان في ما كان في reaction. And then we have allergic contact dermatitis, which is an example of, like, it's just an example, guys, of what is it called? It's just an example of delayed type 4 hypersensitivity. All right. هلا we're gonna go into vaccinology, which is almost the last part. تمام. I hope, guys, that hypersensitivities were clear. If you have any questions, I'll answer them at the end because I really need to finish on time or I'm gonna be killed. أمزح بس إنه. But if you guys have any questions, write them in the chat, okay? I don't want you to forget your questions. Hello. We talked about antibodies like a lot today, like كثير حكينا. This is a slide where guys, you know it. How do antibodies eliminate antigens? So neutralization. So neutralization كيف by basically opsonization. So they kind of neutralization is like they go and they neutralize it. يعني خلاص basically they attach to it and they're like يلا shut up. You know? This is what neutralization is. Phagocytosis طبعا they go and like بيصير عندنا opsonization. So they go and mark the cell and wait for the macrophage to come and engulf it. We have cytotoxicity. طبعا cytotoxicity, this is by natural killer cells. أصلا كيف عرفنا? Guys, how did I know this is natural killer cells? Tell me.
CD16.
صح. Perfect. So there's CD16 and we تتذكروا بال CD16 we said إنه natural killer cells they بيكون في antibodies that are marking, so it's by opsonization. So opsonization can either happen, like it can either help in phagocytosis or in cytotoxicity. Complement activation, we talked about this so many times. هلا let's talk about active and passive immunization. هلا نحن بنعرف إنه في شيء اسمه passive immunization وفي شيء اسمه active. What is active immunization? Active immunization is basically if you are just sitting minding your own business, you got an infection, your sister was sick and she like spread the flu to you. So this is basically your body is actively acquiring immunity against the bacteria because of يعني لأنه أنت you got sick. Okay? So your body actively got it. Or it can be, this is by the natural way of you getting infected. Or active immunity can happen artificially through vaccinations. And what do I mean artificially through vaccinations? We can basically try to mimic the natural way. So how do we get our active immune like system? By getting infection. So when you get infected, your body will attack the virus. It will try to help you clear out this virus. It will try to make you feel better, and it will store memory cells. صح? We're gonna have memory B cells, memory T cells, كل هدول. So we try to put the patient under the same circumstance, and like we expose the patient to an antigen that is related to this like virus or to whatever by vaccinations, and this is considered active immunity. عندنا شيء اسمه passive immunity. By passive, it means that the body is not forming the antibodies. Active means it's actively trying and forming stuff to protect us. But passively, it means we have these things. Like these things are passively given to us. Naturally, these are given by maternal antibodies. So babies at the beginning, their immune system is not fully like formed. فإنه they're protected by their maternal antibodies. That's why breastfeeding is really, really encouraged in the first 3 months. يعني they're always like, I know like they try to like, okay, never mind, this خلاص not important. Next, how do we artificially give passive immunity? By basically monoclonal antibodies, or passive immunization happens by, okay, let's say someone has a disease and it's really, really severe, okay, and we want it to stop. How do we stop it? By basically we provide this patient with antibodies because his body is too weak to form these antibodies. فنحن بنعطيه الantibodies. So you get me? Passive is anything that you give. Passive is anything that you don't make yourself. Active is anything you do yourself. Okay? So passive immunization. So basically, as we said, it's injecting preformed specific antibody. So your body did not form the antibodies. You injected the antibodies that are already formed. كيف؟ ما ضروري تعرف. Antibodies that bind to a toxin receptor. So عندنا something that are, so we have these passive immunization, we have different kinds. We have something called neutralizing antibodies. So remember how we said إنه هون the antibodies can like one of the ways it eliminates antigens by neutralization. So basically it just binds to it and it just tells it shut up. Okay? So this is what neutralizing antibodies means. So these antibodies will bind to the antigen and just like shut it off. We have toxoids. So what are toxoids? Toxoids are From the name, it's like a toxin. You know diphtheria? I'm pretty sure you guys know it. You're literally taking micro. Diphtheria and Clostridium tetanus. Okay? Those two, they are infections that are due to toxins that they're formed. These infections are acquired and the symptoms are there because of the toxins that they release. So you can give something called toxoids. Now, these toxoids, okay, what are they? Listen, these هدول diphtheria or tetani, like Clostridium tetani, both of them, they release bacterial toxins. هدول البacterial toxins, they have two domains. That is really, subhanallah. They have like, okay, this is my toxin, okay? This toxin has two parts. One part is for receptor binding. One part is just the toxic place. تمام? These parts are separated, okay? فهلا أنا, if I want the body to, I want to passively activate the body to fight off this disease. So what will it do? We will give them something called toxoid. What will these toxoid do? These toxoids are gonna go, they're gonna bind to this, and they're gonna just like eliminate it. Okay? They'll bind to the binding domain. Okay? وبس. And the toxoid is usually given to infants because they're not able, like infants at a very young age, they're not able to form like these things, okay? Like now we are able to form things like that, but infants, they're not. Moving on, we have something called anti-venoms, which is basically, guys, you know how if you get like a snake bite and then they give you directly, you go and you get an antidote or anti-venom, this is it. So basically it's things, it's like pre-made antibodies that will prevent the venom from killing you, okay? So this is everything about passive immunization. Is it clear, guys? So passive immunization, anything that is basically preformed antibodies given to you. So they're given to you either to neutralize they're either neutralizing or they're toxoid or they're antivenoms. And you just تفهموا the concept. Once you understand the concept, everything's good. But one thing that is scary, or one thing that we're scared of when we're giving passive immunization, is serum sickness. From the name, serum sickness. So serum sickness is basically a sickness. It's a type 3 hypersensitivity. Yalla, what does type 3 hypersensitivity means? It means بيصير في عنا the antibodies, they're going to bind to an antigen, forming a complex, and this complex is just going to be بنحط everywhere. وبصير في عنا this complex is going to activate the complement. Basically, the complements are going to come and they're going to be like the anaphylatoxins and the شو اسمه الأوبسonins, all of these are going to activate inflammation. So serum sickness, نحن لما نعطي an injection, what happens is maybe our body is going to attack whatever we gave, and they're going to form complex, and these complexes are just going through the blood, and these complexes are going to cause inflammation all over, and it's going to be really bad. So just know إنه serum sickness is something we're scared of when we give passive immunization. تمام؟ هلا active immunization. Active immunization, we wanna stimulate our immune system to produce long-lasting immunity. What are the challenges that we might face? إنه basically مثلا HIV. ليش لهلا نحن we don't have HIV therapy, or why can't we don't have you don't really have like something that can protect you against HIV, right? It's just because for us to produce an antibody response that's really strong, what do we need? We need cell-mediated immune response. When we say cell-mediated, we mean T cells. Remember, guys, in the beginning when we said إنه نحن to provide or to promote or like to have high quality antibodies, we need the T cell to be active, like we need the CD40 to CD40L. So this is one of the things that we face. إنه مثلا HIV لهلا they were not able to form مثلا a vaccine that can stimulate the T cells, okay? Sometimes the proteins alone, like مثلا whatever part you take, هلا نحن when you do vaccines, we're going to talk about the vaccines. We sometimes take parts of the we either take مثلا the virus itself وبنحطه مثل ما هو, we kill the virus or we attenuate it, or we take part of it. Sometimes these parts are not immunogenic. And what do we mean by immunogenic? They don't really stimulate the immune system. But we need to provide something to them, like remember the protein, so the carrier protein, like the conjugate vaccine اللي حكينا عنه بالأول. So we add like a protein to it to stimulate the T cell response. That's the adjuvant right here, okay? Now, هلا نحن when we create a vaccine we need to think of specific stuff, which are these things. So we need to make sure the microorganism does not establish latency. So what do we mean it doesn't establish latency? This virus is protecting the body from vaccines that hide. What are not vaccines, sorry, viruses that hide. What is an example of a virus that hides? Varicella. Herpes zoster, like shingles happen. Why? Because varicella لما انت تكون baby and you get varicella, it might just go and hide in your ganglion, like in your like nervous system, and then suddenly, like later on, decide to reactivate. So when we do like vaccinations against مثلا varicella, we need to make sure the virus, we're not only providing the body with something to prevent the virus from like attacking, or like to protect us from the virus, كمان to protect us from its latency. تمام. It should not undergo antigenic variation. Antigenic variation means it keeps on changing its antigen, and one of the, I don't know if you guys know it, I think you do, one of the most antigenic, like one of the bacteria actually that is very, like it has antigenic variation, and that's why we can't even, like it keeps having a lot of serotypes, is Haemophilus influenza. You guys know it, it's like a gram-negative bacteria. Yeah. Okay. That's why it can be really hard to have like a lot of people can, like all of us can probably got the shot for Haemophilus influenza, but you can still see people who got the let's say epiglottitis or whatever because of Haemophilus influenza. Why? Because it has antigenic variation. It can change its antigen. This is صراحة, it shows how smart this bacteria is. It should not interfere with the host response. This vaccine is only there to amplify the host response. It's only there to improve the response, not to change it. The gold standard is basically to be able to form memory B cells. It's to be able to have T helper cells. So we want to make sure and know we have memory B cells. We want to make sure and know the CD40L happened. So this is the thing that we go over. We want to make sure exactly effective vaccines needs to stimulate both B and T cells. Now let's start with the different kinds. We have live attenuated and inactivated. What's the difference? Live attenuated is so we bring the virus, we either kill it, خلاص it's dead, it can خلاص it's dead, or we attenuate it. And by attenuate it we mean it cannot cause a disease. If I inject it into you, you're not going to have a disease, but it's going to be a little bit stronger than the killed vaccine, because in killed خلاص it's not going to do anything. Okay. بس بالattenuated it's gonna, like the reaction that it causes is stronger. تمام. Do you get me? Because that's why we kind of lean toward attenuated vaccines more than killed vaccines, because okay, killed vaccines are effective. I'm not saying they're not effective, but the immune response that they generate is not as strong as the attenuated vaccines. Why? Because think of it, attenuated vaccines, they are the closest to the real viruses, okay? Because it's still alive, but it's not toxic, بس attenuated vaccines, keep this like احفروها بمخكم, like keep it a rule. You never give it to a pregnant lady or to a patient who has immunodeficiency. Like immunodeficiencies are people who basically, you know what immunodeficiency is. So basically their immune system is deficient. They cannot fight off, like they can't fight off people who are on immunosuppressants. مثلا transplant patients that take medications for life to prevent the immune system from attacking the organ. هدول مثلا. Why? Because these vaccinations, okay, they're attenuated viruses. They're not toxic, but if patients are already vulnerable, the attenuated virus can become an actual virus, or not become an actual virus, but it can be risky. We don't want to risk these patients. Haram, they already have enough. So specifically مثلا the immunodeficient ones and like pregnant lady, you don't want to like affect her baby. Tamam. So this is just, I wanted to show you basically live attenuated, they're more potent and they're better. Why? Because they activate both the B cell and the T cell, and they activate both cytotoxic T cells and T helper cells. Tamam? Okay. Moving on. How do we form? Basically, how do you create these attenuated viruses? So basically, one idea is, I don't know if this is important, guys. This is just a picture in the slide. I just said, why not explain it? It's just that basically, they take this virus, okay? and then they keep on infecting it into يعني مثلا they bring a monkey, they infect this monkey, but then they take again the cells and then they infect them in another monkey and then they infect in another monkey and another monkey, another لدرجة إنه خلاص the virus is exhausted, like it's attenuated, it no longer has its weapons, يعني خلاص it's used all its weapons. تمام. This is how they usually like do the attenuated viruses. A very what is it called? A very important example is the poliovirus vaccine. We have two kinds. We have the Sabin and Salk. Sabin is attenuated, Salk is killed. Sabin is taken orally, and it's طبعا Sabin is more effective, long lasting, and so on, compared to Salk because of لأنه it's attenuated. Salk, it's طبعا killed and it's given by injection, and it needs boosters. Keep this in mind, guys. Killed viruses need boosters. تمام. Now we said إنه نحن we try to improve all of these things. إنه نحن مثلا we like إنه إذا أنت مثلا sometimes if you put the vaccine by itself, like the killed particle or whatever, it might not elicit a strong immune response. So we do something called adjuvants and immunomodulators. This is a photo where you can see, guys, this is a vaccine without adjuvants. So you see how the reaction is. So yeah, like a little bit of reaction. بس شوفوا مع adjuvants, you see how crazy the increase in like activity is. We have more B cells, more antibodies, more memory cells, يعني everything is more. So adjuvants, they're just there to increase the immunogenicity of the vaccine. And we have two examples, aluminum hydroxide and aluminum hydroxide and squalene. So aluminum hydroxide usually boosts B cell response. So if you add aluminum hydroxide to it, it's going to boost the B cell. The squalene, it's going to activate phagocytes. I don't know if you really need to know the details, but just keep that in mind, okay?
Can I ask you a question?
Yeah, sure.
So, for an example, COVID-19, where we were taking second dose active boosters, are they examples of adjuvants where basically we're trying to boost our immune system to produce more T cells and
Yeah, so basically it was probably either a killed enzyme, like a killed vaccine, or an mRNA vaccine. We're going to talk about it هلا بعد شوي. فانه boosters, they just help reminding the immune system, you know, like, oh, this is how you activate me, this is how you do it, okay?
Okay.
هلا moving on, عندنا subunit vaccine. For the subunit vaccine, what is it called? For the subunit vaccine, they take part of a virus and they incorporate it. They don't take the full thing, they take a part of it, a protein. So an example is hepatitis B virus. They take part of its DNA or genes, okay, that basically codes for the surface antigen. What do? They take it, بيحطوها yeast, and then they allow the yeast to reproduce, and they take whatever is produced to form the vaccine. يعني هي basically, think of it this way. They take this gene, they incorporate it here, they increase it, and then the yeast will produce antibodies against this antigen, and they'll just extract it or they'll use it. This is like insulin, guys. I'm pretty sure you took how insulin is made like in high school, and I personally took that. You know, like they just incorporated through biomedical engineering into the yeast, they form it. تمام? Just know subunit is just part of it. هلا delivery vectors, it's just like انه like مثلا نحن when we have a vaccine like an mRNA vaccine. What does mRNA vaccine mean? We take the mRNA and we literally just put it inside a delivery vector and we inject the person. Why? Because the mRNA هو البارت اللي هو immunogenic or هو اللي هيك. تمام؟ فلايك اصلا هون مكتوب subunit vaccines اللي هو basically مثل اللي هلا حكينا عنه انه بتاخدي part of it, بتاخدي part of the DNA or RNA. They require liposomes or basically things to put it. فشايفين هاي مثلا they will form يعني هنا بيحطوا بس يعني مثل مثلا الكوفيد vaccine is a subunit vaccine, by the way. It's like that. They took the mRNA, حطوها جوات like حطوا لها walls and everything when they moved on, you know. هلا في شيء اسمه virus-like particles. It's just like particles from a virus that can cause basically an interaction, you know, like can cause an immune reaction. That's it guys يعني صراحة no need to go into details because there's nothing to say يعني. بعدين عندنا the COVID-19 vaccine, we were just talking about it. So it's, as you can see, it's an RNA vaccine. So basically this is the SARS-CoV mRNA and it's put into a vector, and this vector is what is يعني إنه بنحط يعني this is what is like injected, and the same thing in here. You see this is the virus. They like, okay, wait. They take part of the virus. You see, they take part of the virus, they put it, they incorporate it, and so on. تمام؟ هلا last but not least, it's the last thing اللي هو guys ما بقينا غير like literally for اللي هو basically دكتور نسيت اسمه عاطف عطاكم اياه. Wait, هلا هون we're saying إنه live viral vaccines are better. Why? Because they elicit B and T cell responses. بس طبعا they can be scary, live vaccines. Why? Because they can actually become like, this is the viral, this is, and I'm talking about DNA. ما عم بحكي عن ال attenuated, no, the DNA. The DNA مثلا can be scary because it can cause reinfection, like it can cause the cytotoxic T cells to be activated. تمام؟ هلا هون it's just guys, we said إنه conjugate or like adding an adjuvant is good because it causes linked recognition. So basically, this is the, remember how we said إنه نحنا sometimes the vaccine by like the particle by itself is not immunogenic enough. Okay. Why is it not immunogenic enough? Because of this. Like لأنه basically it's not immunogenic enough. فنحن how do we solve this issue? We solve this issue by binding it to a carrier protein اللي هي البوليساكرايد, which can cause the recruitment of the T cell, وبصير في عنا CD40 مع CD40L وهيك. عندنا something called herd immunity. Do you guys know what herd immunity is? الزبدة تبع herd immunity is إنه basically a lot of people, why do we care if the community gets vaccinated or not? We care because sometimes some people cannot get vaccinated. يعني مثل pregnant people, مثل immunodeficient people, مثل old people. Sometimes they can't be vaccinated. طب how do we protect them? We protect them by herd immunity. Herd immunity means إنه نحن لو عنا 80% of the community is vaccinated against a disease, they're protecting themselves and the people around them. So look at here. So this is no one is immunized. إذا واحد was infected, he went and met all these people, he's gonna infect them all. فشايفين كيف everyone is infected. هون إذا عنا a couple of people that are vaccinated and those people are infected, if they spread the virus, it's less than this, صح? Because we have some people that are protected. بس إذا نحن we increase the number of people that are protected, we're kind of like literally minimizing the spread of disease and protecting people who are vulnerable and they cannot take the vaccinations, اللي هن مثل pregnant people, old people, immunodeficient people. تمام. صراحة guys, you can read this on your own. It's not important. Like we mentioned this a lot. هلا BCG vaccine. هلا we're going to talk about the specific vaccines اللي هن BCG, pneumococcal and meningococcal و HPV, and we're done. Okay. BCG vaccine. Okay. BCG is against TB. هلا زمان it's a live attenuated vaccine. Okay. زمان it was given right at birth, but now it's administered 6 to 12 months after birth. Okay. هلا if it wasn't given, so sometimes parents neglect their kids and they don't give them the BCG vaccine. If it wasn't given in the first year of life, before giving this vaccine, they need to do a tuberculin test to make sure إنه TB is not there, because it's gonna be worse. And it's gonna be useless أصلاً to give the BCG vaccine after a person has already had TB. هلا you have to know إنه BCG only protects from disseminated TB. What is disseminated? The TB that's not in the lungs. So the TB that's in the liver, the body, Pott's disease, all of these, they disseminated out of the body, out of the lungs, BCG can protect from. Anything else, they don't. هلا the issue is إنه why is BCG vaccine really important? It's really important because TB is a disease that is still like, TB is a like يعني an infection that cannot really يعني ما عندنا a lot of vaccine, like sorry, not vaccines, we don't have a lot of drugs that protect us from TB because it's a very hard disease to manage وهيك. the dot therapy, أكيد the doctor told you about dot therapy, where basically they always send people like to the house to make sure they're taking their medications, because you need to take the medications over like months, not only like a week or two. Moving on, عندنا pneumococcal vaccines. For the pneumococcal vaccine, we have pneumococcal vaccine, guys. It's it is one of these, like pneumococcus is one of these, like it's one of these that has antigenic variation, so it keeps changing its antigen. So we have a lot of serotypes, a lot, like عنا أكتر من 100 serotype. بس نحن we care about the things that can cause serious issues like meningitis and antibiotic resistance. So these are the things that we incorporate. We have like a vaccination that is basically PCV that has 13 serotypes. بس والله يعني that's it. No, نحن those 13 stereotypes are the most يعني serious and the things that we need to protect everyone from. طبعا conjugate vaccines offer herd immunity. Guys, this is something we talked about like كتير. Meningococcal vaccine طبعا it's required for Hajj, because when you go in Hajj, okay, you meet people from like everyone all over the world they come, okay. حتى people that are known and they might be carriers, they give them prophylactic antibiotics just to prevent the outbreak of meningococcus. It's usually given 9 and 12 months. Just know these dates, guys, they're important. بس والله they're just saying إنه في عنا new stereotypes that come up. Know the quadruple vaccine? It might be like a question. I don't know, Allah. I remember we got something similar. Just know that it has these 4 because he might ask, like maybe, I'm not sure. Last but not least, HPV vaccine. HPV vaccine is usually given around 11 to 14. But if you guys did not get it up until now, you can take it يعني up until 25 years old, you can still take it. I need to take it يعني. I don't know, like I always get shouted at by doctors and like, oh my god, you guys are educated, why don't you go take your HPV vaccine? For girls, if you didn't take your HPV vaccine, go take it. Boys, same thing, because HPV vaccine can protect from HPV, the cancer that's can cause cervical cancer in females. This is really important, yeah, it's preventable. بس والله, thank you guys for bearing with me. If you guys have any questions, let me know. We can pause the recording. خلاص, end it. Bye guys. If you guys have any questions.